prickly pear. Wow. Okay, let's welcome Trevor to the stage with a round of applause. Is it open? It's not open. Okay, this one. Yes. Where should we stand again? Okay, stand here. Okay, and don't move. Yeah. Okay. Good morning, everyone. My name is Trevor Tsepiso Magonyane, and I'm here to present my study titled Blood Profiles and Performance of Indigenous Non-Descriptive Goats Fed Pringle Pear. These are my supervisors. This will be the outline of my presentation. Okay, this will be my outline of the presentation. Okay, goats are now an important part of South Africa, a South African livestock industry making up to 13% of all livestock in South Africa. Given the significant, blood is an important medium assessing the health status of an animal. Hematological profiling is applied for the assessment of health condition as well as tracking of the nutritional and metabolic status of an animal. Indigenous goat breeds known as paddy goats are particularly significant commercially, nutritionally, and culturally in South Africa. However, during winter and dry season, ruminant productivity in rural areas is low when nutritious feed are unavailable. Hence, it is important to investigate the blood profiles and performance of indigenous non-descriptive goats fed Pringle pear. Such information may be useful to organizations and government aiming at improving the performance and blood profiles in ruminant animals. The aim of the study was to evaluate the effect of Pringle pear inclusion in diet on blood profiles and performance of indigenous non-descriptive goats. And the objective of the study was to determine the effect of Pringle pear fruit inclusion level in the diet on live weight, growth rate, digestibility, diet intake, and blood profiles of indigenous non-descriptive goats. Okay, the hypothesis is aligned with the objective. The study was conducted at the University of Limpopo Experimental Farm in Limpopo local municipality. A total number of 24 non-descriptive indigenous goats were randomly assigned to 4 dietary treatment, completely randomized, to 4 treatment in a completely randomized design, replicated 6 times with 1 goat in each replicate. The data was collected as follows. Pringle pear fruit meal was sourced from a nearby farm, cut and dried as shown in the picture below. Goat blood samples were collected prior to the commencement of the experiment and towards the end of the experiment from each animal using a 5 milliliter syringe and 21 gauge needles. The data was analyzed using statistical package of social science version 29.0. The statistical model was used for mean separation. The Turkey honest significant difference test was used when there were significant differences at 5% level of probability. Okay, under the results, as it shows on here, for initial weight, this is the initial weight, and then where it's highlighted in red, it means that there was an effect in the goats for final weight, average daily gain, and also feed intake. Okay, for blood, there was an effect on lymphocyte, monocyte, neutrophils, basophils, but then didn't have an effect on the eosophil. For discussing Pringle pear inclusion level in diet had a significant effect on final live weight, average daily gain, and feed intake of indigenous non-descript goats. The results were in line with the two authors who reported the sheep gained weight on diets having spineless characters at 50% level of the roughage fraction of the diet, which is higher than the level used in the current study. This might have been due to the increase in diet digestibility with an increase in Pringle pear inclusion level. Blood parameters of indigenous non-descript goats reveal significant difference between the treatment at the beginning of the experiment, as I've indicated. At the end of the trial, Pringle pear inclusion in the diet had no effect on eosophil of indigenous non-descript goats. The results of the current study are in contrary to the results of the author mentioned, who observed no significant difference in monocyte for sheep. The results serve as a baseline to monitor the changes in the blood profile of the goats as affected by the treatments. The major function of the white blood cells are to fight infections and to produce, transport, and distribute antibiotics in immune response. In my conclusion, overall, it is notable that the level of Pringle pear meal inclusion plays a crucial role in determining the nutrient intake and performance of non-descript goats, with 20% being the most favorable based on the results. It can be concluded that the inclusion of Pringle pear food meal in the diet does not have a significant effect on blood parameters. It is then suggested that the Pringle pear meal inclusion levels are used will not cause any adverse effects on the blood profiles of the experimental goats. This will also benefit effect on goat health, which makes it worth and viable feeding strategies. However, it is recommendable that more studies be concluded on the subject to ascertain the present findings. Okay, I would like to acknowledge the Department of Agricultural Economics and Animal Production, and also acknowledge my supervisors and the workers at the experimental farm. These are the references I used for this presentation. Thank you so much.
Thank you, Trevor, for a very nice presentation and for sticking to the time allocated. Okay, and I now open the floor for comments, questions, and recommendations. There is one that said, and you will come later. Okay, please go ahead.
Thank you for the presentation. I just have one question. Given that the prickly pears are wild fruits, how would you advise small-scale goat farmers to actually harvest, or what is your take in terms of them getting the prickly pears and feeding their goats? Should they go there or should they plant? What is your advice in that regard? Thank you.
Okay, thank you so much for the question. Okay, for this study, the first batch of Pringle pear, I bought them, so from a neighbor farm. But the second batch, what I did is that I went to the experimental farm next to the experimental, whereby I got some of them there. So my advice to the small-scale farmers, just to actually make sure that they don't use much funds. They can plant for themselves, because of it's one of the good plants that can actually be found in, especially in dry areas most of the time. So it is found mostly around dry areas. So I'll advise them to actually plant for them to save funds. Thank you.
Thank you very much for the presentation. Mine is a suggestion. And I believe that all of us who are here to better our presentation skills and all that. So my suggestion is that in terms of designing your presentation and all that, I feel like it must be more of points that you can be able to extend on and not read everything that is on the slides.
Okay.
This is not a hard one, but I believe that we are here to learn more on how to present. So next time, maybe putting points and then explain what is on the points, rather than just reading everything that is on the slides. Thank you.
Thank you so much.
Okay, we've got the last two. I think we'll only take 2 questions. And the last one, please, you can engage him during the tea break. Okay, we'll take that one and the last one here. So, thank you.
Thank you for the interesting talk. It's very good to see people trying to explore alternative animal feed. Now, I just have a comment relating to the response that you just provided to the previous learned colleague who asked a question. There are about 29 Opuntia species in the country, and there's about 300 known Opuntia species worldwide. So my first submission is you need to establish which specific species of Opuntia you are using, because in South Africa now there is about 29 Opuntia species, and they are all called prickly pears. So some of them are cactus pears, some of them are sweet prickly pears. So you need to establish which one specifically you are using for your study. And secondly, according to NEMBA, which is one of the regulations, it is classified as category 1B species, which means anywhere it's occurring, it needs to be cleared, eventually eradicated. And therefore, by law, it's not recommended that people plant Opuntia ficus indica. Thank you.
No, thank you so much. Okay, so I think I just forgot maybe to put it there. The species that I use is Opchuga ficus indica. So that's the one that I use. So yes, I'll look into it. Thank you so much.
My question. Thank you, Mr. Trevor, for the presentation. My question is more or less on the potential risk of prickle pear. What did you, to remove the spines? You know, prickle pear has spines. What did you do to remove the spines, like when the goats were feeding on it? Like, what is the procedure behind it? My second one is on its toxicity if it's consumed in large quantities.
Yes.
Like, did you manage the quantities that you were giving the goats? I just basically have those 2 questions for you. Thank you.
Thank you very much for the questions. Okay, on the first question, well, since I was drying them, yes, the spike were there after, but considering that I was mixing them with other feeds, they were not. I took it as if it didn't really affect the goats, because obviously with them, if they go out and then they go graze, they actually eat them like that. So I took it as if, because also when I was drying them, I believe that some of the spines they were actually dying because of I was cutting them, some of them. So, and then on the second question is, I was actually managing that, because of if it's fed too much, it might lead to probably diarrhea or bloating. So what I did is that I did put different levels in terms of the feed ration. Thank you.
We still have 1 minute. There was someone at the back there who wanted to ask a question. Do you still want to go ahead with the question? Okay, thank you. That's it. Let's thank Trevor with a round of applause for a nice presentation. The last speaker for the session is Preeti Rachweni. Her presentation is titled Relationship Between Skin Size, Skin Weight, and Other Related Skin Traits of Nile Crocodile. Let's welcome Preeti to the stage with a round of applause.
Let me first greet the room at large. My name is Preeti Rachweni, here to present the relationship between total skin length, skin weight, and other related skin traits of Nile crocodile. The Nile crocodile, which is scientifically known as the Crocodylus niloticus, are one of the most commonly farmed with crocodilian species in South Africa. Generally, these crocodiles are kept for the production of skin, which contributes for about 80% of the total crocodile skin value, and the 20% is attributed to other byproducts like the meat, oil, and the skeletal matter. The Nile crocodile skin trades accounts for at least 30% of the worldwide skin trade, with South Africa being one of the largest producers. The skins from Nile crocodiles are durable, making them ideal for heavy-duty products like your belts, your shoes, and for those who can afford your Louis Vuitton. However, the crocodile industry is faced with the challenge of producing skins of poor quality and yield, which subsequently affects the price value of the marketed skins. According to studies, it has been stated that the quality and the size of a crocodile affect the market price. And studies conducted in 2004 have mentioned that the quality of the crocodile skin is a crucial component that researchers must look at, as it will eventually impact the quality of the final product. According to a study conducted in 2019, the monetary value of the crocodile skin is determined by multiplying its total length by the price per centimeters, which shows that we need to improve the total length of the skin for us to be able to market the crocodiles at high price values. According to a study conducted in 2021, it has been stated that understanding the relationship between the skin weight and the skin traits would help us improve these variables. However, there is limited literature on the relationship between the total skin length, the skin weight, and the skin traits of Crocodylus niloticus. Hence, the aim of this study was to establish the relationship between total skin length, skin weight, and skin traits of Nile crocodiles, and the objective was to determine the phenotypic relationship of these variables. Hence, the hypothesis is in line with the objective. The study was conducted at Lalele Crocodile Farm in Limpopo Province. The farm is registered with the South African National Standard Crocodiles in Captivity since 2014. A total of 180 skins from 180 Nile crocodiles aged 35 months were used, and the study used a cross-sectional study design with a convenience sampling method, whereby this sample size was decided upon by just convenience to the researchers. This presentation is one objective of an ongoing study which received ethical clearance from the Animals Research Ethics Committee of the University of Limpopo, and that is the certificate number. And the crocodiles were slaughtered at Tabakwena Abattoir, which is registered, which is a veterinary institute registered with the Department of Agriculture, Forestry and Fisheries. Figure 4 just shows a sketch of a slaughtered crocodile, which was made less scary for the sensitive people in the room. If you look at the pictures at the corner there, you can see that is me collecting the traits, measuring them. If you look at the middle picture at the top, you can see that from A1 to A2, that is exactly how the total skin length was measured using a measuring tape calibrated in centimeters. And at the far left corner, right there, that's how the skin thickness was measured in millimeters using a caliper. And right there at the bottom corner, that's me, very excited with a very wide smile there for not being eaten by crocodiles, of course. And at the corner there, that's how we measured the skin weight by weighing it on a scale. The data was analyzed using the statistical package for the social sciences, version 29 of 2022, and Pearson's correlation was used to determine the phenotypic relationship between the total skin length, skin weight, and these mentioned skin traits of the Nile crocodile. If you check at table 1, you can see that on the second column, which is identified at TSL, all the measured traits were correlated except the last trait, which is highlighted in yellow, where the correlation was non-significant. Also, if you check the correlation that's highlighted in red, it shows that the skin thickness was correlated at a p-value of less than 0.05. Also, on the third column, where the skin weight is checked for the correlation with the other traits, you can check that all the other traits were correlated highly. However, the neck total length and the neck width at the middle was not correlated. Therefore, these positive correlations between the variables suggest that if you increase one of the variable, then it will result in an increase with the other variable. The results are in line with a study conducted in 2000 and another one conducted in 2013, which reported that positive correlations between the total skin length and the skin thickness in farmed saltwater crocodile and goats, respectively. The studies, however, in contrast with the study conducted in 2021, which reported that the belly width was negatively correlated to skin weight and total skin length of their Nile crocodiles. These differences, we can attribute them to the different environmental factors. The study concluded that the total skin length had a positive relationship with skin weight and the skin traits such as skin thickness and the other mentioned. However, the neck width from the middle had no relationship with the skin weight. With the total skin length, and the skin weight also had a relationship with a positive relationship with all the skin traits except the neck total length and the neck width from the middle. This means that this mentioned traits that showed a positive correlation may be used to improve total skin length and skin weight of these crocodiles. We recommend that breeders interested in improving the total skin length and the weight may use those traits that showed a positive relationship with the variables. And we also recommend that more research is done using a larger sample size or even different crocodile species. These are some of the references used in compiling this presentation. I would like to thank the University of Limpopo Department of Agricultural Economics and Animal Production for their support, the Lalele Crocodile Farm for allowing us access to their farm, and some of the people who assisted me with data collection, and most but not least, the JASI research team. And you know what they say, in a wild crocodile. Thank you.
Thank you, Pretty, for a very nice presentation and for sticking to time. And I'm now opening the floor for questioning, comments, and inputs. Questions? Okay. 1, 2. Okay.
Okay, thank you very much with the nice presentation. I just want to ask maybe a question to say, did you consider other factors such as age and gender of those crocodiles?
Okay. Like I said, this is part of an ongoing study. So on my current study, one of the objectives is to check the effect of the sex and age. However, we did not necessarily consider the age when selecting the skins. So we just used all the skins that were available, regardless of the gender. Yes, thank you.
Thank you, madam, for the insightful presentation. My question is on your discussion. You mentioned that the differences may have been attributed by environmental factors. What are those environmental factors? Thank you.
Okay. Like I said, when I was doing the study, it was at Lalele Crocodile Farm, but the study which also used the same breed was in other farms. So some of those environmental factors may be the feed that they provide the animals, it may be the temperature in the enclosures, it may even attribute to maybe how the animals are managed just generally. So it's one of those factors, just the areas that they are kept in and the areas that the skins from this current study were kept in. Yes, thank you.
More questions, please. More questions? Okay, over there.
Thank you for your presentation. My question is a little bit out of what you are investigating about. So I'm more interested in like where does the meat go? Maybe it can be an alternative source of protein or some other nutrients. Where does the meat go after you slaughter your crocodiles and harvest the skin?
Of course, of course. The meat plates actually contribute for about 15% of the production of crocodiles. So there are some people who like produce the animals basically for meat. But yeah, also here, if they are slaughtering the crocodiles, they also be able to store the meat and then they sell it. There at the farm, there's actually some sort of a restaurant, I don't know if I can call it, where they sell the meat themselves. So yes, you can eat the crocodile meat, and it's known to be high in the fatty and the omega vitamin for those who knows those sources. But yeah, it can be an alternative source of protein, especially in this economy of poor food security. Yes, thank you.
Okay, that's it. Let's thank Pretty for a very nice presentation with a round of applause. And that marks the end for this session. And I also request a round of applause for having managed the session very nicely. And we can now break for tea. Thank you very much.
Good morning, colleagues. Good morning. Good morning. Yeah, no, it's Mongalo. I think it's this one. This one. This one. This one. Yeah. This is the correct one. Yeah, sure. It's fine. The person will go ahead. And the person will do it. The better way. Oh, the better. Colleagues, welcome back. Colleagues, welcome back. Let us continue. We don't have time. My name is Andreas. I'll be driving the next session. I repeat, my name is Andreas. I will be the driver of this session. And our first presenter will be Ruben Mongalo. He will be telling us about the investigation of adopting ICT technology among small-scale farmers, a case study of Limpopo province. Is the person here? Let me remind the presenters that they are still having 12 minutes to present and 3 minutes questions or inputs. Thank you very much, sir. You are protected.
Good morning, everyone. I'm Mongalo Ruben, and I'm here to present about the title of my studies, Investigating Adoption of Information Communication Technology Tools by Small-Scale Farmers in Blouberg Local Municipality, Limpopo, and supervised by the following supervisors, Zwane and Mr. Mpatlele. Here's the outline of my presentation, starting with the introduction and ending with the references. Based on the introduction, agriculture is a pillar of many developing countries and developed countries by ensuring that it measures global food security and economic stability through fostering the small-scale farmers. As small-scale farmers, they are a crucial component of agricultural production in many regions. Also, it also contributes to the rural livelihoods of farmers and also rural-based people. Yet, small-scale farmers face various challenges, including limited to resources, more like input resources, and also the ones that financial resources, where farmers don't get sufficient support to invest in their daily day-to-day farming practices. The study is also going to talk about mostly the ICT tools that is going to address it: radio, television, smartphones, and landline. Yet the problem statement, as it is, the main source currently of agricultural information to farmers is extension service, which is a major challenge for information to be delivered to farmers efficiently, as one extension officer is assigned 850 farmers. Yet now there are recent studies they're suggesting that the one extension officer must serve 250 farmers. Narrowing down the ratio will ensure that all farmers receive information so that they can practice whatever they practice in their farming fields. And also, based on the adoption rate, small-scale farmers remain subject of concern and investigation, as some of these concerns relate to low level of education and poor adoption of technology, as some of the technologies, they are very complicit, and most farmers in rural areas, they are not digital literate. As like it becomes a major problem for the farmers to adopt digital tools, because of like most they lack education, like they lack digital literacy, so it becomes a major problem. That's why there's a low level of adoption of ICT tools for farmers to access information. On these challenges, they emphasize the need for hybrid methods by agricultural extension and also by other stakeholders, so that they can improve the efficiency of information and transformation of agricultural information to farmers. As we are moving into the fourth industrial revolution, most farmers, particularly the small-scale farmers and emerging farmers, they will be left behind due to lack of digital literacy. And also, one of the major issues of this thing of adopting ICT tools by small-scale farmers is a problem of electricity. As like it is well known, like currently in our country, we're having a problem with electricity, power shortages, mostly known as load shedding. So during like where the information must be transmitted to farmers in rural areas, when there is load shedding, most of the network towers where like farmers or people have to connect to internet, there's a problem of network. So it results in a way like flow of information that's supposed to get to farmers is disturbed because of poor infrastructure. On the rest, now small-scale farmers in South Africa could benefit more to use of ICT tools, as like ICT tools, they make sure that farmers get what they need at their own comfortable space, more like information, particularly information relating to whatsoever, like mostly the information relating to production information and also management information, and most importantly, where like farmers can ask their extension officers about certain things that they don't understand or the issues that they're experiencing in their farming fields, and also where farmers can receive information relating to financial support, where they can apply so that they can get assistance from Department of Agriculture and also from several stakeholders. ICT initiatives boost the rural poor's overall being, while also generating significant agricultural investment in rural areas through fostering of ICT tool in agriculture. It helps to improve the rural livelihoods of farmers and also people who are based in rural areas through where they can access information so that they can farm and also produce, when also like it will also measure the major concerns that we're having, like of national food security and stability, where farmers can just produce and have access to nutritious food. And the study, like, is also in relation to this, the extent of ICT adoption in Blouberg local municipality is currently unknown. The study also seeks to address the following questions, which is like, what are the key socio-economic factors that influence the initial adoption of ICT tools by small-scale farmers, and also which factors influence the access and adoption of ICT tools in different farming practices in Blouberg local municipality. The aim of the study was to investigate the adoption of ICT tools that can be used or adopted by small-scale farmers, and also here below like their objectives, where the objectives of the study, like, one of them was to determine socio-economic factors of small-scale farmers in Blouberg local municipality, and the last one was to determine factors that influence the adoption of ICT tools in farming, different farming practices. As mostly known, not each and every practice where like we have livestock and we have crop production and we have poultry, where like we can use the same ICT tools to use in our day-to-day operations. The methodology, like the study like was conducted and using this, the study used the quantitative approach where like for the researcher used the questionnaires to collect data. Even though like the researcher anticipated to collect 110 questionnaires from the participants. Due to unforeseen circumstances, like the researcher only could collect only 69 research questionnaires through the participants, as some were due to lack of, due to transportation costs, and also those areas that they were remote. So it was a major problem for the researcher to go and collect data there. In relation to analytical tools, the researcher used descriptive analysis to measure the first objective. Then the second objective was measured using cluster analysis. And thirdly, which is the last one, was measured using ordinal regression analysis. Below, like the results have revealed that ICT tools investigated were radio, smartphone, computer, and television, and landline. Among all the investigated ICT tools, radio was the one showing stable patterns, indicating that adopters and non-adopters, they still prefer radio as the traditional source of information. Because the study also revealed that particularly the study is dominated by people who are over the age of 50, and also like those people lack formal education, which makes it understandable because of like what? Due to challenges of education, like where most people didn't receive formal education, it made it difficult for them to adopt or use other ICT tools like smartphones. All age and education and farming experience contributed to the influence of ICT tools to the graph, to this graph, where like the high spikes of the graph. Well, on the blue line, it's usually the adopters. The orange graph is the non-adopters. The first point, which is radio, is showing stable patterns where like farmers, like I also have said, or like most of them they were lacking. The influence were due to lacking of formal education and also age, being older people. On the graph also, like smartphones shows like it's dropping a bit. It may be due to lack of affordability, because as we know, most like data like is currently expensive, and also due to connectivity issues. The study it has revealed that the necessity to adopt hybrid strategies to improve farmers' digital skills and also foster youth into agriculture, so that in the near future the scale or the level of small-scale farmers can be upgraded due to the fostering of youth into agriculture, as youth are mainly active, can also understand the use of digital tools. Like my acknowledgements, I would like to acknowledge AgriSeta for funding, and also Prof. Mbengwa and Prof. Zwane, and also University of Limpopo for allowing me to undertake the study as my reference, and thank you.
Thank you very much. Thank you very much with a nice presentation. Questions, I'll allow only 3 questions. If you have 2 questions, you ask one. Then the rest will engage with the presenter during lunchtime. 1 question per person.
Sorry, this is not a suggestion. It's a question. It's just a suggestion. You have a line graph, okay? It should be a bar graph, because those things is not connected with each other. It's different sections that you are showing which one is the best or whatever. So the one is not connected to the other, connected to the other. So rather use a bar.
Okay, thank you.
Thank you very much. That's for him. There are no questions. The next speaker will be contribution to crop performance and yield. Please clap hands for her before she can arrive on stage. Thank you very much, ma'am. You can proceed.
Good morning, everyone. My name is Refilo Matata, and I'm here to present the study titled Tillering Propensity and Tiller Contribution to Sorghum Performance and Yield Parameters as a Function of Cultivar Selection and Varying Irrigation Regimes. Now, sorghum is a drought-tolerant crop which has a yield-adaptive trait to water stress known as tillering. Now, the tiller propensity tends to increase with increasing water stress. Now, this tiller fertility has a range which is from 7 to 91%, which correspond to a yield contribution from tillers ranging from 5 to 78%. The tillering propensity is known to operate through a complex interaction of genetics, environmental conditions, and the management factors, which then lead to difficulty into quantifying the contribution of tillers to yield under the interaction of varying cultivars and different irrigation regimes. Up to our knowledge, no scientific study have quantified the contribution of tillers to yield given the interaction of water availability and varying sorghum cultivars. Now, since tillers are known to contribute to yields, it is then important that we provide empirical evidence based on the tiller success to farmers, which will help them in selecting the cultivars based on their given water availability scenarios, which will help in optimize and increase the tiller contribution to overall yield as a tool to improve food security. The study was aimed at providing empirical evidence to showcase the contribution of tillers to yield under the interactions. And we with the objective saying the effect of varying irrigation regimes on tiller propensity, performance, and yield parameters of sorghum cultivars. The study was conducted at the University of Limpopo Experimental Farm, laid out as a split plot using a random RCBD with replicated 3 times. So the study comprised of 3 irrigation regimes, which is full irrigation, deficit, and also rain-fed, having 4 sorghum cultivars, namely Pan 606, Mr. Basta, Marcia, and Elenrace. Now the irrigation were carried out, the full irrigation was carried out weekly at 80% field capacity throughout the growing season, whereas deficit irrigation was carried out from the flag leaf stage, which is the stage prior to the flowering, which is the most sensitive stage for sorghum, till physiological maturity, and also at 80% field capacity. So for the rain-fed plot, the crops depended solely on precipitation. The soil samples were collected for soil characterization. These parameters are needed to be inserted in the SPAL model and to give the readings of field capacity, which aided into the irrigation. Now, the tiller ranking and tagging was done on 10 plants, where the main stem was tagged M, when the first tiller to appear was tagged T1, and so on. So the soil water content was collected weekly prior to irrigation. The flowering and physiological maturity was also collected. The biomass was collected where the above ground, where the shoots were destructively harvested and then they were dried. The yield, the panicles were then threshed for the grains, and the harvest index was determined. The data collected was subjected to analysis of variance using GenStat Vision 20. The means were separated using LSD at 5% significance level, and multiple mean comparison was done using the Benforoni. Now the results here indicate that the interaction of irrigation regimes and the cultivar selection significantly influenced the shoot-dry biomass, whereby I then indicated that the land race had significantly higher shoot-dry biomass compared to Mr. Basta. This high shoot-dry biomass could be attributed to the plant height or the plant height and the plant stem width, where land race as a tall cultivar, it then have a large leaf surface area which allows them to intercept more light and aid into photosynthetic, leading to high biomass. Now the contribution of tillers to shoot-dry biomass. Graph A indicate the contribution within the stem ranks, where main stem had indicated to have contributed higher shoot-dry biomasses compared to the tillers. This could be attributed to main stem being the first shoot that appears after emergence, and which then stand a higher advantage of getting more resources. So for harvest index, the main stem and tiller one had indicated to have a significantly same harvest index, which is higher compared to tiller 5. This could be due to main stem and tiller 1 being synchronized in their phenological development, where they reached flowering at the same time and also physiological maturity. Now this one here tells us that more yield, more biomass was then partitioned into the harvestable parts. Therefore, it then shows that we expect more yield from the main stem and T1, not from the T5. Now the contributions to harvest index it still shows that the main stem is the major contributor too. So now here under the different cultivars, also the main stem is the larger contributors. However, under irrigation regimes, under deficit and full, it is then shown that the main stem was not the major contributor. This could be attributed to tillers taking charge into compensating for yield in case the main stem failed. For yield, the results indicate that the interaction of cultivar selection and irrigation regimes significantly influenced the grain yield, where under all the irrigation regimes, land race had indicated to have higher yield. The high yield of land race, the high yield compared to Mr. Basta. This high yield of land race could be attributed to it being a traditional variety, a local variety, and also having the propensity to tiller higher compared to other cultivars. However, Mr. Basta also, it's a high tillering hybrid. However, here we had low yield for it just because the tillers could have engaged, they would have engaged in the competition between the stem ranks where they competed for the resources, which then resulted into lower yield. So the the percentage of fertile tillers indicate that Mr. Basta had higher number of tillers. However, the competition that way comes in. However, under the fertile tillers, it shows that most of them were not fertile. That's why we have less. So for irrigation regimes, deficit had indicated to have high yield compared to other irrigations. Now the contribution to grain yield between the stem ranks differed significantly. Main stem also here did show that it had a higher contribution compared to other stem ranks. Now the under cultivar selection, since tillers are known to contribute from 5 to 78% to yield, here we used 5% as the cutoff for significant contributions. So here it shows that the land race ahead up until T3 that is harvestable. Same applies to Marcia, Mr. Basta, and Pen. So under different irrigation regimes, all the irrigation regimes also indicated that T3 was the harvestable, was the cutoff for the harvestable, for the harvestable yields. So in conclusion, the interaction had showed to have significant difference. Therefore, it is a suitable strategy that farmers can employ in order to optimize yield from the tillers. And then, with that being said, we have quantified 57% overall tiller contribution from the yield under the interaction of cultivar and the irrigation regimes. So we now recommend to the farmers who wish to cultivate under deficit irrigation that they should opt to cultivate landrace and pad, as they have indicated to have at least acceptable yields under this water regime. Whereas for full irrigation, Massia indicated to thrive better compared to other cultivars. Now, for farmers to target the harvestable tillers, the cutoff is at T3, since it has been indicated that T3 is up to till 3, we have the tillers, we have the yield that is contributing. Thank you.
Thank you very much. Thank you very much. Questions? One question per person. Thank you. There's a hand here. There's another hand. We have 2 hands. You may start.
Thank you for the presentation. So my question is on the irrigation regimes. I saw that on your grain yield, deficit irrigation was high, even better than the full irrigation. So, but I'm interested, how did you practice the deficit irrigation?
Okay, thank you for the question. So, the deficit irrigation, I've mentioned that it was the crop was supplemented irrigation from the flag leaf stage, which is from 45 days after planting. Therefore, that plot also indicated to have taken time to reach maturity. That's why it had high yields under that. So for deficit irrigation, it differed from full irrigation, because full irrigation is when we applied throughout the growing cycle of the crop. Whereas for deficit, there were certain stages that did not receive water at all. That's how we practiced it. I'm not sure if I did answer your question.
Thank you, madam. I really liked your presentation. So on your I think it was second slide, you presented a hypothesis. So, and you mentioned that your result was significantly different. So I wanted to know, from your p-value, would you accept or reject your null hypothesis? Thank you.
Given the p-value, we reject the null hypothesis. Thank you. Oh, I did. Just that I forgot to. Yeah.
Thank you very much. Clap hands for her. The next speaker is Nelisiwe Matlangu. There's a slight change in the program. She's supposed to be presenting in the afternoon, but she asked to be squeezed for this session. Nelisiwe Matlangu. Please clap hands for her before she can arrive.
Greetings, everyone. My name is Nelisiwe Mahlangu. Under the supervision of Professor Bopape Mabapa MP, I'll be presenting on the growth, yield, and nutritional composition of Moringa oleifera under different propagation methods. Moringa oleifera is the most extensively grown species in the Moringaceae family, which is rich in essential nutrients beneficial for human health and also valuable for addressing nutritional deficiencies in most developing countries. Moringa oleifera is propagated sexually through seeds and vegetatively through stem cuttings, and improving the propagation techniques can increase moringa availability. The limited access to a healthy, balanced diet is the major problem in most rural communities, and the high food costs and reliance on social grants restricts access to nutritious food. Moreover, the increasing population threatens food security, leading to malnutrition, and despite agriculture being the primary livelihood, most of the hungry individuals are from farming families. So these communities show interest in cultivating the moringa tree, but there is a lack of awareness and the knowledge about its cultivation. The development of moringa requires careful plant propagation to provide a high quality seedling, since the tree is well known for its use in the production of nutrient-rich food and other products, and moringa has a great role in food security and can improve the livelihoods of smallholder farmers. Therefore, its production will contribute towards food security and income generation. The study aimed at investigating the effect of propagation methods on growth, yield, and nutritional composition of the moringa tree, and recommending the best method that can be used for propagating it, with the objective to determine the best propagation method or methods that can maintain high growth and yield of the moringa tree and high nutritional composition of the leaves, with the hypothesis that says propagation methods will have no effect on growth and yield of moringa tree and in maintaining the high nutritional composition of the leaves. The study was conducted at the University of Limpopo Aquaculture Research Unit using the randomized complete block design comprising of 3 treatments, namely the direct seeding, seedling transplanting, and the stem cuttings, each replicated 6 times. The polyethylene bags were used in the study, and they were prefilled with topsoil collected from the study area. The figure 2.2 shows the experimental layout with the plot size and the spacing between the treatments. For direct seeding, mature seeds were collected from healthy pots at the University of Limpopo Experimental Farm, and the seeds were soaked in tap water for 24 hours to aid water absorption and sprouting, and 3 seeds were sown directly into each polyethylene bag at a depth of 2 centimeters, and thinning was performed to leave 1 healthy vigorous seedling per plot. For seedling transplanting, 2 seeds were sown per plastic bag prefilled with propagation mix. Seedlings were irrigated every 2 to 3 days in a shade house, and 1 week after emergence, the seedlings were thinned to retain the strongest one. The seedlings were nursed for 8 weeks, reaching a height of 15 to 30 centimeters before being transplanted to ensure better survival rates. For stem cuttings, the clones were harvested from branches with developed hardwood, and the cuttings were prepared focusing on the position and direction of the cup-like nodes. Each cutting was approximately 45 centimeters long, and the tip portions were discarded and leaves were trimmed to promote sprouting. For data collection, the following parameters, the growth and yield parameters, were collected, and for nutritional composition, the following nutrients were analyzed using the methods mentioned on the slide. The data collected were subjected to analysis of variance using GenStat 18th edition, and means were separated by the least significant difference at a probability level of 0.05. Figure 4.1 shows the daily emergence rate of Moringa oleifera influenced by different propagation methods. The graph indicates that seedling transplanting demonstrated the quickest and most reliable emergence rate, likely due to the pre-established root systems of the seedlings, and the stem cuttings also had a good emergence rate, but slightly slower than the seedling transplanting. Then the direct seeding resulted in slower emergence rate. This could be attributed to the time required for the seeds to emerge and establish. Figure 4.2 shows the plant height of seedlings influenced by the propagation methods. The graph shows that direct seeding resulted in shorter plants across all the time points, and the stem cuttings resulted in taller plants compared to direct seeding, whereas the seedling transplanting resulted into the tallest plants and showed significant difference from both the direct seeding and the stem cuttings. These results agree with Jacob et al. 2016, who argued that stem cuttings have a superior genetic makeup than seeds because seedlings produced from stem cuttings had a greater mean height than seedlings produced from seeds. The figure 4.3 shows the number of branches per plant influenced by different propagation methods. The graph shows that seedling transplanting produced a significantly higher number of branches compared to the stem cuttings and direct seeding methods. These results contradict with Olorukoba et al. 2030 on the influence of plant methods on growth and vegetative yield of Moringa, who revealed that the number of branches were not significantly affected by planting methods. Figure 4.4 shows the chlorophyll content influenced by the propagation methods. The graph shows that chlorophyll content was highly significant in stem cuttings only at week 28 after planting. This could be due to the controlled conditions in the shade house that offer optimal development opportunities, leading to the improved chlorophyll production in seedlings that continue to grow when transplanted. And Monisha et al. 2019 demonstrated that propagation methods did not yield notable effects on chlorophyll content. Figure 4.5 shows the Moringa biomass production as influenced by the different propagation methods. The graph shows that seedling transplanting resulted in higher biomass, although it was not significantly different from stem cuttings, and direct seeding resulted in significantly lower biomass compared to the other methods. Table 4.1 shows the effect of the methods on micronutrient. So the iron and zinc were highest in stem cuttings, followed by direct seeding and seedling transplanting being the lowest. Then manganese was highest in direct seeding, with stem cuttings and transplanting having the same value. Then table 4.2 shows the effect of different propagation methods on the macronutrients content of Moringa. The table shows that there were no significant differences in macronutrients of the tea. However, the potassium showed higher content in direct seeding. Seedling transplanting led to faster growth rates and higher yields compared to direct seeding and stem cuttings, and these results contradict the initial hypothesis that propagation methods would have no effect. The findings emphasize the importance of choosing the right propagation method to enhance moringa growth, yield, and nutritional quality, and given its effectiveness, there is a strong recommendation to promote seedling transplanting as the preferred method for cultivating Moringa oleifera, and farmers and practitioners should be encouraged to adopt these methods for optimal results in terms of growth, yield, and nutritional content. I would like to acknowledge the University of Limpopo Food Bev Center and my supervisor. These are the references that were used in this presentation. Thank you.
Thank you very much. Thank you very much. Questions? 1, 2, 3. Thank you. Start.
Greetings, everyone. Mine is not a question, but then it is a suggestion. You have very interesting data and you have obtained relevant data. So my suggestion is that you can use other statistical analysis techniques such as multivariate. With multivariate, you can clearly show the interaction of variables with, which can greatly contribute to broader understanding of the effects of variables without only being tied to the differences of variables. So I believe in your case you can use the CPA, which is the PCA rather, which is the principal components analysis to clearly show the interaction between the variables. Thank you.
Thank you so much for your suggestion.
Please, when you're asking a question or suggesting or putting inputs, let's stand up a little bit. Yes, let's stand up so that we can also be on the TV. Thank you.
It was a suggestion. Yes. You got it right.
Yes, thank you.
Thank you, Nelly Sue, for the nice presentation. I just wanted to find out, since this study is mainly focusing on the farmers that are rural based, like if I got you correctly, how would you ensure that your findings from your study are effectively put into use within the rural areas? Because there's, you mentioned about nurturing seedlings for 8 weeks and all those. How will you make sure that all that knowledge is delivered to those people that are in the rural places and put into use effectively? Thank you.
Thank you for your question. This study is gonna be published, so that's how I'm gonna ensure that the results are effective to the farmers in rural areas.
To be for public consumption, to be published, be for public consumption.
Thank you for your presentation. My question is, you mentioned that Moringa oleifera can be propagated using seeds. So I wanted to know what were the best environmental conditions which were appropriate for the growth of your seeds.
Can you repeat the question, because I can't hear you clearly?
Okay,
Speak
you
louder.
mentioned that your Moringa oleifera seeds can be propagated. Your plant can be propagated using seeds. So I wanted to find out what were the best environmental conditions appropriate for the growth.
Okay, thank you for your question. So the appropriate conditions for growing moringa, the tree requires the hot environment, I don't know if I answered your question correctly. It requires the hot environment and also, you said the environmental conditions, right? Then it also requires the soil that is sandy, it has good aeration, and yeah.
Thank you very much. Let's clap hands for him. The next presenter, or his presenters, for the first time ever in this kind of an event, we are going to have two presenters simultaneously. The other one will take the first part, the other one will take the second part. The students are Chaba M. Please let us give them the hands of applause before the time. They will tell us about the following topic or title: Good farmers' perception of drought impact and local adaptation measures in Polokwane municipality. Goat farmers. We'll hear about the team. Thank you very much. Presenters, you are protected.
Greetings, House at large. I go by the name of Chaba MR. I'm not alone, I'm with Mr. Develo KP. Today, welcome to our presentation. The title is Goat Farmer Perspective of Drought Impact and Local Adaptation Measures in Polokwane Municipality, Capricorn District in Limpopo Province of South Africa. Here is my outline. Problem statement. Goat production play a crucial role in improved livelihoods and food security in many small farmers, particularly in rural area of South Africa. The current drought in South Africa impacts fodder and freshwater availability and consequently threaten goat production. Majority of local smallholder farmers are well aware of this issue, but are unaware of the drought adaptation and mitigation strategies. To mitigate this problem, it is necessary for farmers to find alternative drought-tolerant feedstuff, such as prickled pear. The picture below shows prickled pear. Studies conducted by Cordova 2022 prove that prickled pear is as well adapted to drought, and it contains 80 to 90% of moisture content. Moreover, it is rich in energy content. Our aim is to investigate goat farmer perspective of drought impact and local adaptation measures in Polokwane Municipality, Capricorn District in Limpopo Province of South Africa. Our objective is to evaluate the local adaptation measures in Polokwane Municipality, Capricorn District in Limpopo Province of South Africa, and strategy currently employed by goat farmers to mitigate the effect of drought. Our objectives are aligned with our hypothesis. Study site. The study was conducted in 3 villages, Radihale, Chweni, and Maja village of Polokwane Municipality, Capricorn District in Limpopo Province of South Africa. Research design. Cross-sectional study design and data was collected via questionnaire, which were administered once to each farmer of non-district indigenous goat. Sampling procedure and sample size. The 3 villages were purposely selected with 24 farmers with at least 10 nondescript goats from each village. Thus, 72 goat farmers were interviewed. Data collection. Data was collected through face-to-face interview using word structure and questionnaire. Data analysis. Statistical package for social science version 28.0 software was used for data analysis. Ladies and gentlemen, I will hand over to my colleague to continue with the results and discussions. Thank you.
Good day, ladies and gentlemen. I'm Tabele KP as introduced, and then I'll carry on from where my colleague has left. The table presented on the slide indicates the demographic characteristics of these farmers that were interviewed from these villages. And then we noted that 81.9% of them were males rather than females, and then which is in agreement with the results of Onzima et al. And Sheriff et al., who reported that majority of goat keepers are male farmers. It was very necessary to, find out about the goat breeds that were kept in these villages, and then the mixed goat and the paddy goat accounted for the highest percentages of these breeds that were kept in these villages, in contrast to Boer goat, which is the least goat breed that is kept. And then these results are also in agreement with the results of the stated author, who observed that most goat farmers prefer to farm with local breeds because they are better adapted to the environment, rather than introducing an exotic breed to the environment. And then these are the pictures of the goat breeds that were kept in the villages. The first one is of the Boer goat, and also the second one is Savannah goat breed. We interviewed these farmers for their reasons to keep goats, and then we found out that 86.1% of them keep goats for generating income, and then followed by 42% which keep goats for traditional purposes, such as, malobola, they lobola, yes. And then the least, or let me rather say, the last reason for keeping the goats were for consumption, which accounted for the least percentages. And then these results are also in agreement with the results of the stated author, who observed that most farmers keep goats for consumption and also for investment. The following 3 slides are kind of related, hence they complement each other. The first one being the feeding practices, which refers to how these goat farmers feed their goats during dry and wet seasons. As depicted by the graph, I can't point there. Most farmers, majority of them, they allow their goats to free roam in the environment in both wet and dry seasons. And then some of them, they tether their goats, which is they tie them in a pole so that they don't go far. And then this is in agreement with the stated author, who found that free roaming was the primary feeding practice for goats in both dry and wet seasons. And then related to that is why the fodder in indigenous trees and also grass account for the highest percentages as feed resources for goats in these villages. And then the last one that is related to the two is feed supplementation. We found that most goat farmers, they don't supplement their goats. They rather use traditional trees, and then the least of them, they purchase feeds, because finding is a major problem in the rural areas. And then this is in agreement with the stated author, who indicated that shrubs and tree leaves as supplements yielded positive results in good performance. We later on introduced a strategy that can be used to mitigate the drought implications, which is feeding goats crops that are well adapted to drought, such as prickle pear. And then, unfortunately, we found out that many of these farmers, they don't know that prickle pear can be fed to goats, which is why 93% they said they don't know that prickle pear can be fed to goats. And then, this is the reason why most of these farmers, they don't know the effects of prickle pear to goats. But if I may go back, the 4.2% that said they allowed their goats to feed to prickle pear, they said that they noticed increased weight gain and also they noticed higher immunity to diseases. This is in agreement with the many authors who found that livestock fed cactus pear improved their body weight. And then we found out about the impacts of drought and methane, which was not part of the study. But at least all of these farmers, they know about drought, just that they don't know about the mitigation strategies to alleviate the effects of drought. However, majority of these farmers reported no implications as a result of drought to goats, which is contrary to findings of Alabadi, who found that drought has numerous negative effects and consequences in rural communities, which is why we conclude from this study that farmers are very much aware of drought, but they lack knowledge on how to mitigate drought. And then farmers are also, they do not know the effective coping mechanisms of drought. And then they are aware, the awareness of prickle pear as an alternative feed during drought is very much lacking, which is why we appeal to government also to intervene in terms of training workshops and drought management and livestock care, so that farmers can be well aware of other feeds that can be fed to goats to mitigate drought and to mitigate the effects of drought. These are the references that we used to compile our study, and then I would like to thank University of Limpopo and also the NRF for funding this study. Thank you.
Thank you very much for good presentation. Unity. Questions? One, two, three, because we are two, I'll add another, the last one. Four questions. One question per person, please.
Thank you for the presentation, guys. I'm reading the topic of your research, and I also listened to what you presented and what you did, and I'm trying to understand where the element of perception of drought impact and local adaptation measures, where that really comes in, because you did something, the topic says something, you are reporting on something. It's like oil and water. I'm not really getting what you did.
Okay, thank you for the question. The purpose of the study was to know whether the farmers from rural areas, they know about drought and also how to mitigate the effects of drought. You understand? That is the main purpose of this study. So which is why we interviewed them as indicated by the last slide there. Which one? This one. And then we asked them questions about drought, we asked them questions about methane, and then majority of them found out that they know about drought, but they just don't know about the how to mitigate these effects of drought. I don't know if I answered your question very well. I didn't.
I think even on the objective, there was no element of even myself, I quoted it, there was no element of perception at all.
Perception.
Yeah, it was not there on the objectives, but it's there on the A.
Right, let me try to go back to objective. Oh, okay. I'll, okay.
Thank you for your presentation. One thing that I've realized when you're presenting is that you said these farmers, they are aware, but then they lack strategies, you know, to mitigate these drought impacts. So I want to know, what do they do during drought seasons? Because you never reported on their mitigating strategies. What do they do?
Okay.
Like during drought season, do their livestock die off, then the next season they start again from, yeah, so that's it.
Okay, one of these strategies that we noted that these goat farmers use, whereas it's few of them, they buy commercial feeds, you know, and then many of them, they just allow their goats. As I mentioned, they've little to no knowledge about the strategies to mitigate drought, but fortunately, many of them, they provide water to their goats. They have boreholes, you know, and I think it's one of those strategies that we can use to mitigate drought. But if I may tell you, they don't know about also prickle pear. They don't know about prickle pear. They don't know how to mitigate this drought impacts. So I found out that one of the best ways to mitigate a drought is by reducing the stocking rate of animals, which they don't know about. They just keep their animals until the next season. Did I answer your question?
Thank you.
Thank you. Most of the presenters today are presenting on prickly pear, and Dr. Moshobani has highlighted the fact that the prickly pear is an invasive species and they are in the process of trying to eradicate them. So I want to ask you that you mentioned that they do use some of the indigenous plants to feed their goats. Is there no alternative, maybe that is more or less similar to the prickly pear that they can use to feed their goats?
If I may try to understand your question, you are trying to ask, since the government is trying to eradicate prickle pear, what are other alternatives for mitigating this? Literature says that, as I said when I was answering that question, they must reduce their stocking rates, either by weaning earlier. Yeah. Oh, they are there. The likes of lucerne, they also can be fed to goats. But then these plants, they need to be bought. So that is why we try to, advise farmers to adapt to plants that are locally available that doesn't require funds or that doesn't require to be bought. So I would mention lucerne as one of those plants and crops that can be fed to goats and also other livestock. Thank you.
Okay, you're saying they are there, but they're expensive.
Yeah, they are there. Very good.
Question.
Okay, thank you for your presentation. I'm not sure if you can go back to your aim and objectives slide.
Yeah.
And then you said your aim was to investigate the drought impact on local farmers. So how does you checking the gender of the people who are fast goats and their mode of feeding relate to your aim?
Well, the concept of demographic characteristics was just to find out and also compare to the other literature review that's about the majority of farmers, whether are they male or female. It's kind of not related to the aim, but it was very much necessary to find out, because as we were interviewing these farmers, we found out that many of them are males, as I've also indicated on the slides, but it is not related to this. It was just to back up the research. Why did we, investigate these farmers?
Yes. If you're not satisfied, you're going to lunch with them, you sit at the same table, you eat together, you ask other questions. Thank you very much. Clap hands for them. The next speaker is Tabang Mamavulo.
She will talk about small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change. A case study of Makuru Tamara, local municipality. Please clap hands for her. This is not a teamwork. She's alone. Only 3 questions will be allowed. Thank you. Ma'am, you are protected.
Good day, ladies and gentlemen. I am Tavang Mamabolo, and I will be presenting a study co-authored with Dr. M.A. Nkwana titled Small-Scale Maize Farmers' Willingness to Pay for Changing Planting Dates in the Face of Climate Change, a case study of the Makudu Tamara local municipality, Limpopo Province, South Africa. Before going to the problem statement, may I kindly inform you that for the context of the study, willingness to pay for changing planting dates refers to the extent in which small-scale maize farmers are willing to sacrifice to change planting dates, and does not necessarily have a monetary value attached to it. However, it does have an economic value attached to it. This is a presentation outline. Maize is an essential grain classified as a warm weather crop that can be grown in diverse environmental conditions. It forms part of our daily diet as a staple for human consumption and feed for livestock, especially in rural communities. According to Nda, Weby, and Woolway in their respective studies, climate change introduces vulnerabilities to the agricultural system, decreasing maize output by 30% and feed for livestock. These vulnerabilities are faced by countries relying on primary sector economic activities due to increased production and vulnerabilities to the agricultural system, decreasing maize output by 30%. As thus, small-scale maize farmers experience low production productivity, income, and increased transaction sector economic activities due to increased production and has the potential to reduce the negatives of climate change and its variability on small-scale maize farmers experience low production productivity, income, and increased transaction promote sustainable welfare of the disadvantaged. Climate change adaptation has the potential to reduce the negatives of climate change and its variability on small-scale maize farmers, especially in rural communities. It also has the ability to reduce vulnerabilities and promote sustainable welfare of the disadvantaged small-scale maize farmers. Stakeholder participation, which includes researchers, policymakers, agricultural extension officers, service providers, and local communities, is deemed essential for the successful adoption of adaptation strategies in agriculture. Studies by Bain et al. 2012 and Park et al. 2013 have been conducted to identify different climate adaptation strategies. However, the extent to which small-scale maize farmers are willing to pay for changing planting dates is not well established in the study area. Hence, the aim of the study was to analyze the socio-economic factors influencing small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change in the Makudu Tamara local municipality of Sekhukhune district, Limpopo province. The objectives of the study were to profile socio-economic factors of small-scale maize farmers in the study area, as well as to determine factors influencing small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change in Makudu Tamara local municipality, Sekhukhune district of Limpopo province. The hypothesis of the study stated that socio-economic factors do not influence small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change. The study was conducted in the Makudu Tamara local municipality of the Sekhukhune district, and small-scale maize farmers were possibly sampled, wherein 150 maize farmers were sampled using the probability proportional to sample size sampling method. This is the number of small-scale maize farmers that were sampled from 3 villages in the Makhuduthamaga local municipality. Primary data was used to collect, was used as a form of data collection in the use of structured questionnaires which had open-ended and closed-ended questions. Descriptive statistics was used to address the first objective and the probit regression model, the second objective. SPSS version 28.0 was used to analyze the data that was collected. This is a table showing the description of continuous and categorical variables that we used in the study. Displayed on the screen is a table of descriptive statistics which shows the frequency table that shows the minimum, maximum, mean, and standard deviation of the population that was sampled. On the side is a bar graph showing the employment status of the small-scale maize farmers in the local municipality. These are pie charts which show results that were obtained from the descriptive statistics, that is the educational level of the small-scale maize farmer, use of indigenous forecast, source of income of the small-scale maize farmer, and exposure to climate information services to the small-scale maize farmer. These are the results of the probit regression model. From the 12 variables, only 7 were found to be significant. Years of farming experience was significant at 1% level, with a negative effect on farmers' willingness to pay for changing planting days, in the face of climate change. With marginal effects of 0.01, there is an implication that a unit increase in the years of farming experience decreases the probability of the small-scale maize farmers' willingness to pay for changing planting dates by 1.1%. The level of income was also statistically significant at 1%, with a positive effect on small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change. With marginal effects of 0.28, this implies that a unit increase in the small-scale maize farmers' level of income increases the probability of the small-scale maize farmers' willingness to pay for changing planting dates by 28%. Therefore, income level influences willingness to adopt climate adaptation measures. Again, exposure to climate information services had a statistical significance of 10%, with a positive effect on farmers' willingness to pay for changing climate change. It contributes positively to farmers' willingness to pay for changing planting dates, where the marginal effects imply that a unit increase in exposure to information services to the small-scale maize farmers increases the probability of the small-scale maize farmers' willingness to pay for changing planting dates in the face of climate change by 27%. The study therefore rejects the null hypothesis and accepts the alternative, which states that small-scale maize farmers in the Makhuduthamaga local municipality are willing to pay to change planting dates in the face of climate change. Intervention from the South African government on climate awareness and exposure to climate information strategies could assist small-scale farmers to be able to curb any influence or effect that climate change might have on their production and profit. Government and support intervention through provision of incentives and subsidies could aid these small-scale maize farmers to help them get more income as they use this small-scale maize production as another source of income generation in their households. Provision of agricultural training to small-scale maize farmers could also assist small-scale farmers be able to put to practice what they have been taught in these workshops. Government officials can assist small-scale maize farmers through climate change awareness and educate them about the weather patterns and climate change adaptation strategies. Therefore, the study recommends that there should be policies designed to address the dissemination of climate information among small-scale maize farmers. Also, service delivery should be tailored to meet the needs of different socio-economic groups of small-scale farmers in the rural areas for adaptation purposes. Acknowledgements to the co-author of the study, Dr. M.A. Nkwana, the Makhuduthamaga local community, the University of Limpopo, and the Faculty of Science and Agriculture. These are the references that were used. Ladies and gentlemen, thank you.
Thank you very much for a good presentation. Questions? Questions? 1, questions, 2, questions, 3, 1.
Thank you for the presentation, Ms. Mama Wolo. So I'm just interested, what is the current maize planting date in South Africa?
The current maize planting dates in South Africa, planting starts in October to mid-December. However, things have changed based on how climate change has affected production. So I would say it depends on the region at which the farmer, the small-scale maize farmer is at, because our climate and temperature conditions are not the same.
Thank you.
Thank you. So I think my question will be more of a follow-up question. So looking at those planting dates, maybe let's say a farmer is situated in a semi-arid region. So how would you advise that farmer, like in terms of changing the planting dates? Should they plant earlier or later? Looking at, considering the change in the patterns of the climate precipitation and all those climatic conditions.
Thank you for your question. Based on how climatic conditions and temperature are constantly changing, I would advise a small-scale maize farmer to start their planting mid-November, looking at the fact that in December, we get a lot of rainfall. So in October, chances are that the seeds might die off. They may become dormant due to little to no amount of rainfall, unless if that farmer has their own source of irrigation, which they can use to be able to water their plants. Also, I will encourage that the use of fertilizer, pesticide, insecticides be reduced, as this also has an impact on climate change and on climate change.
Thank you very much. Clap hands for her. The next speaker before lunch, this one is in between. You are between lunch and people. The next speaker is Thabo Ntswane. Thabo Ntswane, who is here? He will present on the multivariate-based optimization of green synthesized zinc, a case study of Limpopo province. You are in between lunch and people, please. Thank you very much. You are protected.
Thank you. Good afternoon, everyone. I am Ntswane Thabo, and I will be presenting to you multivariate-based optimization of green synthesized zinc oxide nanocomposite for the remedial of ampicillin in aqueous solution under the supervision of the following. My outline is presented as follows. Ampicillin is an antibiotic which belongs to the penicillin group. It is predominantly used to preserve human health and animal well-being. However, of recent, it has been viewed as emerging pollutants as its release into the environment is increasing. Therefore, antibiotic-resistant bacteria and antibiotic resistance genome are some of the known effects of this ampicillin. These pollutants are persistent and resistant to traditional treatment processes. Therefore, consequently, nanotechnology is viewed as a better removal of these pollutants with the use of nanoadsorbent due to increasing their removal efficiency. Nanoadsorbent have been synthesized using different approaches. However, due to their limitations, green technology is the most preferred method. This is because the method is eco-friendly, environmentally friendly, and biocompetitive. The method involved the use of plant extracts, enzymes, and microbial polymers such as fungi and bacteria. Now, in this study, the bacteria will be used to synthesize the green nanocomposite. The aim of the study is to remove ampicillin from synthetic water using green synthesized zinc nanoadsorbent. This aim will be achieved under the following objective, which is one is to extract the biopolymer from Providentia hexaensis, two, to synthesize and characterize zinc nanoadsorbent, to optimize the synthesized zinc nanoadsorbent using chemometric approach, and lastly, to remove ampicillin from the synthetic water. Now, the precipitate method was used to obtain the crude biopolymer from the bacterium, and that the procedure involved the inoculation of the bacterium into the media and cultured at optimum conditions. Therefore, the crude biopolymer will be centrifuged, shaken, and left to precipitate, whereby the precipitate will be vacuum dried to obtain the biopolymer. Now, the ex situ synthesis method was used to synthesize zinc nanoadsorbent using the biopolymer and the zinc oxide solution. The mixed solution was subjected to heating, centrifuged, and vacuum dried to obtain zinc nanoparticles. Now, various characterization techniques were used to study the functional group on the surface of the nanoparticle, to establish the crystalline nature, and lastly, to ascertain the morphology of the particle. The experiment was run in few stages, whereby in the first stage, based on design of experiment, Box-Benken design was used to generate a number of a set of 20 tests which are going to be run under varied conditions. Optimization, which is the second stage, based on response surface methodology. Second order polynomial model was used to fit the data for predicting the optimum conditions. And lastly, verification of the model was done in the lab. And these are the parameters which were optimized. Now, a maximum yield of 3.5 grams per liter was observed. These results were considered best as compared to other studies in literature, observing a yield of less than 3 grams per liter using different microorganisms. Now, the biocomposite present in the biopolymer were responsible for capping, reduction, and stabilizing of the nanoparticle. As indicated in the figure, the biopolymer and the zinc nanocomposite exhibited similar functional groups. Moreover, these secondary metabolites are able to bind to metals and will alleviate any aggregation, thus resulting in highly stable nanoparticles. Now, SEM analysis revealed the zinc nanoadsorbent to be irregular hexagonal shape. 5 major peaks were observed for zinc oxide, whereby they corresponded to the crystal wizard structure, and that the biopolymer and the zinc nanocomposite exhibited similar peaks of less intensity as compared to the zinc oxide. This signifies a successful synthesis of the biopolymer, of the nanocomposite, excuse me. Now, adsorption is mainly affected by several factors, whereby dosage, increase in dosage result in an increase in the removal efficiency. This is attributed to the fact that increased dosage result in increased surface area for the ampicillin to bind to. Secondly, an increase in concentration up until 35 milligram per liter resulted in an increase in the removal efficiency. Thirdly, pH increase resulted in a decrease in the removal efficiency. This is attributed to the fact that a change in pH of a medium result in a different structure of the nanoparticle and different ionization of the ampicillin. Therefore, an increase that is greater than 4 pH resulted in negatively charging the structure of the nanocomposite, thus resulting in a negative charge which will repulse the ampicillin, resulting in a decreased removal. Now, counter and response surface plots were used to investigate the interaction between factors. Therefore, 2 factors were varied at a time while keeping the other constant, and that the green shaded region illustrates the highest removal efficiency. And in this case, concentration and dosage were the most significant factors. Now, the highlighted parameters resulted in a predicted yield, a predicted removal of 101.48%, and that a laboratorial experiment resulted in a peak removal activity of 97.36%. A D of equals to 1, as observed on the figure, indicated desirable response. In conclusion, a biopolymer yield of 3.5 gram was successfully extracted from the bacteria, and that it was used to synthesize zinc nanoparticles, which were verified using the various characterization techniques. Moreover, Box-Benken design based on response surface methodology observed a removal activity of 97.36% under the stipulated conditions. Therefore, in conclusion, the ampicillin was efficiently bioremediated through subjecting it to green synthesized zinc nanoadsorbent. I would recommend that more characterizations such as thermal stability, degradation, and size of the nanoparticle are necessary for clear understanding of the nanoparticles and that we also recommend the scale up of the nano-adsorbent and its application in real water, wastewater application. Now, I'd like to acknowledge the following. The following are my references. Thank you.
Thank you very much. Quick questions. 1, 2, closed. Straightforward question is a direct order. Sate. You can start. It's no problem. You can start.
Thank you very much, and interesting talk, I must say. Especially if we were to talk about nanotechnology, which is of course what I'm working on. And, but I have a couple of questions, but as I'm told I have to ask one question, let me be very specific and straightforward. And let me correct you before I ask.
No.
The usage of weights. You're talking about nanocomposites, you're talking about zinc oxide nanoparticles, so I get confused. What are you talking about? Nanocomposites and nanoadsorbents are two different things. That is the first thing. The second one, that was a comment. The first question, just one question. Which biopolymer did you use? Thank you.
I used Profidentia hexaicens. Yeah. I obtained it from the bacteria.
Thank you. Dr. Sati?
Sure, thanks very much. I see that you used the box design, but you have about 4 or 5 factors. So my question, because they said it must be quick, I thought you were going to use central composite design. Why did you use the box? Because you have 4 factors.
Okay, so with the box bankend design, you can also use up to 4 factors as minimum. Yeah, it will generate a 20 number of runs. But however, in this case, as compared to the central composite design, I think box bankend design has more numbers of runs.
Thank you very much. Clap hands for him. And please clap hands for me. Thank you. Announcement. Those who are going to present in the next session after lunch, please, before you go that side, upload your slides. That is the first thing. Secondly, we are having lunch for 55 minutes. 55 minutes. 55 minutes. We are not leaving there at 55 minutes. We should be here at 55 minutes. It means you are leaving that side. We are having only 50 minutes.
Ashava, okay, and then Trudy Monwa. Oh, okay. So our presenters, remember you have got 12 minutes to make your presentation and 3 minutes for questions. And I would also like to request that when you are busy presenting, please look on your right-hand side. We'll be showing you the minutes you are remaining with. So allow me to call the first presenter, Tato Seleka, who will be taking us through exploring socio-economic determinants of goat head size, a case study of smallholder farmers in Lephalale local municipality, Limpopo, South Africa. Can we please give her a round of applause?
Good afternoon, everyone. My name is Tato Seleka. I'm here to present a paper titled Exploring Socio-Economic Determinants of Goat Head Size, a case study of smallholder goat farmers in Lephalale local municipality, Limpopo, South Africa, co-authored by Professor MP Signolo and Dr. JP Mukaukau. Goat farming is one of the largest agriculture industries in developing nations, Africa being a home of about 35% of the world's total goat population. The value of goats stems from the fact that they may be milked for domestic use as well as for sale. Other socio-economic benefits of goats are religious, festive, and ceremonial situations. Small livestock head numbers restrict farmers' ability to commercialize their head and capitalize on the rising demand for livestock and livestock products. In South Africa, a larger portion of goat population is maintained in poor conditions, and some of the goats are left to fend for themselves in poorly maintained grazing lands. And again, they are not even given feed supplements. Smallholder goat husbandry usually lacks veterinary care, resulting in a high mortality rate, subclinical illnesses, and low reproductive rate. While purchases are a primary source for the initial stock, births are the principal way for increasing the head size. High mortality rates are the major outflow channel through which livestock leaves the head. And reducing mortality rates has a positive effect on the livestock population, as evidenced within the goat subsector. The commercial sector has been significantly impacted by growth of livestock, which has mostly concentrated on boosting productivity and income. The advantages of the unorganized rural goat sector, however, have been limited. The comparative limited number of goats that each smallholder farmer or household has is one of the primary causes of those limited advantages. This study purpose was to highlight the focus on socio-economic factors influencing the goat head size among smallholder goat farmers in Lephalale local municipality. The aim of the study was to analyze the socio-economic factors influencing the head size among smallholder goat farmers in Lephalale local municipality. The objectives of the study was to identify and describe socio-economic characteristics of the smallholder goat farmers in Lephalale local municipality, and to analyze the socio-economic factors that influence the head size of the smallholder goat farmers in Lephalale local municipality. The hypothesis of the study was aligned with the objective. The study was conducted at Haselega village of the Lephalale local municipality under Waterberg district municipality in Limpopo province of South Africa. A simple random sampling technique was used to select 61 participants in Haselega, and primary data was collected through a semi-structured questionnaire. To select the sample size of 61 participants, the Yamani sampling formula was used to calculate the sampling size from the estimated population of 159 farmers in Haselega. Descriptive statistics was employed to address the first objective, and a multiple linear regression model addressed the second objective. This is the model equation. The results for descriptive continuous variables are as displayed on the screen. The study found that majority of the farmers were elderly, and the education level was that most of the farmers ended school with the secondary level education. The distance to the nearest market ranged from 40 to 50 kilometers, and the mean was 44.59, which states that, or which simply imply that farmers had to travel considerate distance to the input market, and this can positively affect the goat productivity, since staying next to the input market can lead to the farmers reducing the cost of transportation. The years of experience in farming range from 2 years to 36 years, with the mean of 13.64, approximately 14 years. This states that most farmers were experienced, and this could lead to the positive productivity of goat, because the more the experienced the farmer, the larger the head size, since the more experienced farmers know how to deal with challenges is within the goat husbandry. And the head size, which is the continuous variable, was and the head size, which is the dependent variable, ranged from 5 to 31, with the mean of 13.72. These are the other socio-economic characteristics of the farmers. The result shows that most of the farmers were males and married and unemployed. The other socio-economic statistics of the farmers are displayed in the table. For total household income, no farmer reported that they receive less than 1,000 rand, and majority of them reported that they have the total household income between 1,000 rand and 5,000. This could imply that the more the household income, the larger the head size. Because if the household income increases, farmers are able to spend for their expenses and also opt for buying other goods to expand their heads. Pertaining to access to output market, access to credit, and access to veterinary care. Not access to veterinary. And agricultural training, no farmer reported that they received that in the past 12 months. Only 91.8% of the farmers did not receive the veterinary services for their goats. And for reasons of farming, the majority of the farmers stated that their reason for farming was for consumption and marketing. The results for the model are as shown. Out of the 11 tested variables, only 7 of the variables were significant. The gender level of education, the household income, the employment status, distance to market, years of experience in farming, and reason for farming. In conclusion, a lack of veterinary services, poor farming skills, poor housing facilities, market issues, a high incidence of diseases and pests, and a high cost of animal medicines, and a lack of feed constrain smallholder goat farmers in the Lephalale local municipality. The study showed that there are several factors that influence the head size among the smallholder goat farmers in the Lephalale local municipality. So the null hypothesis, which stated that there were no socio-economic factors determining the head size among smallholder goat farmers in the Lephalale local municipality, was rejected. For recommendations to secure increased output and productivity, the study recommends making significant efforts to educate and retain knowledgeable goat farmers, because education is important for farmers and also for the community. The prospect of providing training on scientific rearing practices suited to their style of rearing and the scope of generating additional income to prove their livelihood can be looked forward to in future upliftment projects. Since the employment status were found significant, it is advisable for policymakers to make some programs that may assist unemployed or underemployed individuals in goat farming, providing them with incentives and resources for their head expansion.
Thank you, Tato. Thank you. Can we clap hands for him? I will only take one hand due to time. Tato took her time. So only one burning question.
Thank you so much,
Thank you, Tato, for that. I'm asking because no one is willing to ask, so that at least we can put some marks together. You make some random statements that I'm not so sure where you are getting your answers from. Because when I looked at what you were asking and the questions and what you assessed, for example, one of your last comments was increased output, and I didn't hear you, during the presentation saying anything about those farmers needing to increase the herd. Then you talk about the more the income, the bigger the head. The more the household income, the bigger the head. I didn't find anywhere where you can prove that statement as correct. Then you also said the more the years of experience, the larger the herd. And such statements, and I couldn't pick up where you are proving those things from based on your questionnaire and what you were investigating. Thanks.
For household income, I believe it was found positive and significant. And this may imply that when farmers have more disposable income, they can spend for their expenses, and with some other money, they can opt for buying other goods and expand their heads. And this study, these results are in line with the study of 4 et al. 2018. And for years of experience, I believe that when farmers are more experienced, they are familiar with many challenges that are faced in the goat husbandry. And them being familiar with those, they can easily tackle them whenever they face them again, or whenever they appear again.
Okay, thank you, Tato. Please let's clap hands for her. Then without waste of time, our next presenter is Theophilus Mashava, who will be taking us through assessment of ranch land health and resilience in Blowback local municipality using MODIS and DVI. Can we please clap hands for him? Please check our timekeeper as you're presenting. Remember, you've got 12 minutes.
Greetings, everyone. I go by the name of Theophilus Temban Mashava, and today I'll be taking you the presentation under one of my objectives for my research, which is the assessment of rangeland health and resilience in Blaubeck local municipality using the MODIS and the VI time series. Rangelands are landscapes that are primarily dominated by woodlands, shrublands, and also the wetlands. In arid and semi-arid regions, these vegetation have a socio-economic value as they serve as the critical element for biodiversity conservation, the carbon sequestration, the soil stabilization, and also for food security. In South Africa, these resources, they cover more than 70% of the land surface. However, there are a number of factors which are affecting these rangelands worldwide. These include also here in South Africa, which includes the soil erosion, the biodiversity loss, and also most seriously, the climate change. According to the recent estimates, the global temperature is estimated to be increasing by 1.1 to 6.4 degrees Celsius by 2100. Limpopo province has been identified as one of the most affected provinces in South Africa, which is affected by the climate change, both in terms of the variation in extreme event and average temperature and rainfall pattern. As a consequence, it has become more crucial to comprehend how climatic factors like temperature and precipitation affect vegetation productivity. This study assessed the state of rangelands in Blaubeck local municipality in Limpopo province, South Africa, focusing on their vigor and the ability to recover after disturbance. The study has been conducted within the Blaubeck local municipality, which is approximately 95 kilometers northwest of the Polokwane city. The study area is located within a semi-arid region, which has a warm summer seasons and dry winter seasons. Due to the Suspense-Back mountain location, the region expresses seasonal drought and also the orographic rainfall between May and October each year. The study area receives a total average rainfall of 410 millimeters and between 16 to 40 degrees in terms of temperature. The assessment of rangelands has been done using two different methods, which include the field work, which requires more time and it costs effectively. However, satellite images has emerged as a powerful tool for monitoring vegetation dynamics across global ecosystem, offering a cost-effective and time-effective data acquisition. The MODIS and the VI of MODI 13Q1 dataset has been used for these analysis purposes. This dataset provides a 16 days bi-weekly NDVI value at a spatial resolution of 250 meters above ground. The MODI 13Q1 of Vision 6 dataset was imported directly from the Google Earth Engine data catalog using the JavaScript code. This Google Earth Engine was chosen for its powerful cloud computing capability and vast archive of remote sensing data. The GEE was accessed via the attached link, this one. The NDVI and the EVI bands were extracted. These are the most useful vegetation indices to monitor vegetation health. Each of these vegetation indices contain values ranging from -1 to 1, where the higher values indicate more health vegetations. To provide more clear picture of vegetation trends over time, a bi-weekly vegetation indices value were aggregated into annual averages. For the analysis purpose, a pixel line regression model was applied across the vegetation indices time series to detect the long-term trend in vegetation health and the resilience, where a positive slope indicating improved vegetation health and the negative slope indicating the declining vegetation health over time. The results were further visualized to show the spatial pattern in vegetation dynamics across the municipality, with red color highlighting areas of degradation and the green color highlighting the area of resilience. The first diagram, it shows a significant year-to-year variability in terms of vegetation in terms of NDVI values reflecting a fluctuation in vegetation health and a potential environmental factors such as rainfall pattern and land use changes. We can note that 2014 and 2021 and 2022 shows a marked increase in NDVI, indicating a period of recovery or a favorable condition for vegetation growth, with 2022 recording the highest NDVI value within the time series. While there are fluctuations, there are general trends from 2001 to 2022 shows a modest overall increase in terms of the NDVI value, suggesting a possible improvement in vegetation health or better land management practices over time. The variability and recent upward trends in NDVI could indicate a resilience in rangeland vegetation, but significant fluctuation highlights vulnerability to environmental stressors, which could be further investigated for better management strategies. The next graph, it shows the mean annual EVI from 2001 to 2022. The Enhanced Vegetation Index is designed to improve the sensitivity of the NDVI in areas with higher biomass by reducing the atmospheric influence and minimizing the canopy saturation. However, in areas with sparse and degraded vegetation, the EVI values are generally low compared to the NDVI. The consistently low EVI values compared to that one of the NDVI across the 2001 to 2022 period could indicate that the study area has relatively low biomass or sparse vegetation cover, which limits the EVI upper potentials. Similar to the NDVI values, or similar to the NDVI graph, the EVI graph shows the fluctuation in vegetation health over time, with sharp drop in 2003, 2010, and 2012. These drops likely to correspond to dry periods or other harsh environmental conditions. We further analyzed the spatial pattern of the municipality. Looking at the spatial trend, there is a general consistency in NDVI pattern over the 22 years period, with the central green area, which indicate a high NDVI value, maintaining the relative stable vegetation health, while the outer region shows more variability. The green belt seen consistently in the central region across all years, highlighting an area of persistent vegetation health. The temporal variation in NDVI likely reflect the impact of climate variability, with low NDVI years correlating with potential drought events and high NDVI years with wetter conditions, emphasizing the role of climate in shaping the vegetation health. The outer area shows more fluctuation in NDVI over the years, suggesting that these regions may be more sensitive to climatic change or land use practices such as the grazing or agriculture compared to the more resilient central region. In summary, the findings, to summarize the findings, the observed trend suggests that the rangelands have undergone both the period of decline and also the period of recovery over the past two decades. The climatic condition, the land use, and also the management practices could be the factor which is influencing the fluctuation in the vegetation indices. The positive trend suggests that the rangelands are becoming more and more resilient and recovering from previous period of stresses. This study underscores the efficiency of using the Moody's NDVI for a long-term rangelands monitoring, offering a valuable insight for land managers and policymakers. By identifying regions of concern and tracking over time, targeted intervention can be implemented to enhance rangelands health and their resilience. I would like to acknowledge the University of Limpopo, the National Research Foundation, and also the Center for Global Change at C-Block in the University of Limpopo. This is the reference that I've used. I thank you.
Thank you, Theophilus, for your presentation. presentation. Any questions that we have for our presenter?
Thank you for the presentation. What's happening at that green belt on your figure?
Sorry?
The green belt that's consistently on your diagrams, what is happening there?
This one.
Yeah, what is that place?
This is, it highlights an area where there is a possibility that they are practicing more, they are managing their rangelands in a better way. It indicates the vegetation health. It shows that the vegetation health, the vegetation is more healthier than the surrounding areas.
Yeah, I know. What I'm saying is, in reality, did you ever check what that area is, where that area is?
No.
Okay, thank you. One last question. Yes, you are muted.
It's on, yeah. Okay. So, in the beginning of the presentation, you spoke something about the time series, and then you went on to present. Descriptive data, right? So my suggestion is just that maybe after doing all the descriptives, I think you must also have more focus on the inferential side, or more focus on the inferential, getting more of inferential results than just the descriptives, because I think inferential results gives you more chance to conclude or give overall conclusions than just analyzing just the trend in a graph and assume that something is happening. But if you can do more of inferential statistics results, you would be able to see different effects in data. Thank you very much.
Thank you. It's noted.
Thank you. I hope that you managed to get the comments and you'll definitely effect them. Let's clap hands for him for the presentation that he has gave us. While we are calling forth Trudy Monam, who will be taking us through performance and carcass traits of Ross 308 brother chickens fed varying 309 levels. Let's clap hands for her as she is coming. To the presenters, please, when you are presenting, try to be stable. Our virtual family, they are struggling to follow. Let's try to be stable when we are on the podium.
Good afternoon, everyone. My name is Mona Trudy. I'll be taking you through my research titled Growth, Performance, and Carcass Traits of Indigenous Bushveld Chicken Fed Varying 309 Levels. My co-authors are as follows. This is the outline of my study. Smallholder farmers in Southern Africa rely on indigenous chickens for economic and dietary protein as they are disease resistant and culturally they can survive in rural areas. Despite the lower productivity, indigenous bushveld chickens are favored for their meat and fit for the demand of organic and free additive meat. The high cost of conventional protein sources such as soybean in poultry that hinders the cost-effective feed formulation, especially in indigenous chickens. 309, the third limiting amino acid in broiler diets, play a vital role in poultry nutrition, impacting growth and productivity, with feed costs compromising 80 to 90% of production cost. Therefore, the study is aimed at evaluating dietary 309 levels for optimal production and carcass characteristics of indigenous bushveld chicken. The study was conducted at the University of Limpopo Livestock Unit. Indigenous bushveld chicken were purchased at Bushveld Range, whereas ingredients were bought at Simple Grow Agri, Meadow Feeds, and Organics Meta Company. And on the other hand, disinfectants, light and wood shavings, medicine feeders were bought at NTK. In this study, 75 male chickens were used in completely randomized design, having 5 dietary treatment levels replicated 3 times with 5 chickens per replicate. 309 inclusion levels were at 5 treatment that are as follows. Freshwater and feeds were offered ad libitum with 24 hours of lighting. During the day, it was natural lighting, and at night, it was artificial lighting. Live weight feed intake and FCR, meat parts, meat color, and sensory evaluation and cooking loss data was collected. Therefore, the data was subjected to one-way ANOVA using statistical analysis. On table 1, it shows the effect of 309 inclusion levels on feed intake, body weight, and feed conversion ratio of male indigenous bushveld chicken. So on the table, it shows that 309 inclusion levels had an effect on feed intake, body weight, and FCR. Then on table 2 of the effect of 309 inclusion levels on meat parts of male indigenous bushveld chicken, it shows that all parameters were affected by the inclusion of 309 levels. Then on table 3, which is the effect of 309 inclusion levels on breast color and pH of male indigenous chickens, it shows that meat breast color was affected, whereas the meat pH was not affected by the inclusion of 309 inclusion levels. Then on table 4, the effect of 309 inclusion levels on meat sensory attribute and cooking loss of male indigenous bushveld chicken aged 91 days. It shows that 309 had an effect on the tenderness only. It had an effect on the tenderness. It had an effect on tenderness, whereas on the other hand, juiciness, flavor, and overall acceptability and cooking loss were not affected by the inclusion levels of 309. On discussion, 309 levels impact feed intake, body weight gain, and feed conversion ratio in male bushveld chicken aged 50 to 91 days. This agrees with the study of Valizade et al. 2016 chalked, who reported that 309 inclusion levels affected feed intake, body weight gain, FCR of broiler chickens. However, this result contradicts earlier findings on optimal 309 levels for feed intake and body weight gain in different chicken. For carcass traits, higher 309 levels at 9 grams per kg increased carcass, drumstick, breast, and thigh weights, showing positive relationship between 309 inclusion levels and the other parameters. The study's findings on carcass and meat parts, color, pH, and cooking loss and sensory attribute align with some literatures of LEME and NCR, but are in contrast with others of Faram. So in conclusion, the dietary 309 levels significantly affected the various parameters such as feed intake, body weight gain, FCR, and meat quality in bushveld chicken aged 50 to 91 days. Optimal 309 levels varied by parameters with specific needs for body weight gain and abdominal fat identified at different levels. So it is recommended that diet formulation should be tailored to the breed and parameters of interest, considering that the 309 requirements may differ from other broiler lines. These are the references used to compile
this presentation. I would like to thank NRF for funding this study. Thank you.
Thank you, Ms. Mona, for your presentation. We only have time for one question, one burning question. Okay, in the absence of questions, let's clap hands for him for the presentation that he has made. This was the last presenter for the session that I was chairing. I was told that we are going to have a virtual presentation, yes, in few minutes. Yes, thank you. Can you clap hands for me as well?
He's coming.
Oh, he has to enter things. Oh, it was not connected to the meeting.
I wanted to ask for the pronunciation of his name.
What is the name? Sandile. Sandile Motsa.
So it's Motsa.
Yeah, Motsa. Yeah, ask him. It's fine. Oh, okay.
Yes, so it's Motsa.
Yeah, Motsa. Is it off?
Yeah. No, yeah, mine it's never.
You put it on when you get home.
Yeah. It's never. I'm worried. Where are the people? It's just, and the cameras will be here. These chairs where we're not seeing people. Perhaps where is he?
I can hear you. That.
The camera. Put it on so that
You can see me. And then after that I'll close it, right?
Yeah.
And also the mic.
Yeah.
Okay.
And also the mic again when he speaks. Yeah, I think you're good to go.
Okay. Good afternoon and welcome back. Noise. Behind the boards. Good afternoon, colleagues, students, ladies and gentlemen. Thank you for getting back on time. And we are now ready to listen to our international keynote speaker. As I've indicated, it's Professor Sandile Mutsa from the University of Eswatini. First, let me thank Professor Mutsa for making it possible for us to reconvene this way so that he can address us. So I'm not going to waste any more time. Then I will read his short biography. Professor Sandile Mutsa is currently serving as the Dean of the Faculty of Science and Engineering at the University of Eswatini. He holds honorary professorships at the University of KwaZulu-Natal, Wits University, and is a professor extraordinaire at UNISA. He is a distinguished applied mathematician, a founding member of the Kingdom of Eswatini Academy of Science. He specializes in the development of numerical methods for solving mathematical models in science and engineering. Professor Mutsa has over 200 publications and more than 5,000 citations. He is an influential researcher and serves on the editorial boards of prestigious journals. He has contributed significantly to the Southern Africa Mathematical Sciences Association as the Vice President and as the President. He has supervised numerous postdoctoral fellows, doctoral and master students. Professor Mutsa is passionate about integrating artificial intelligence and technology into education and research, regularly sharing his insights through his column, which is named Artificial Intelligence Corner in the Eswatini Observer. So ladies and gentlemen, he will be presenting his work under the following title: Leveraging Generative Artificial Intelligence to Transform Mathematics Education, Innovative Solutions for Complex Problems. Can we please give Professor Mutsa a round of applause as he gets ready to talk to us? Thank you, Professor Mutsa. The time is yours.
Thank you. Can I just confirm if you can hear me clearly?
Yes, you are.
Okay.
We can hear you.
Yes, thank you very much, and thank you for the introduction. And I'd like to thank the organizers of this event for extending the invitation to me to participate in the Research Day activity. Let me just share my screen and so could you confirm if you can see the screen clearly?
Yes, we can.
Okay, thank you very much. Then I will proceed. You will alert me if there are issues. As I have been introduced, I will be sharing insights on how artificial intelligence has transformed the teaching and learning of mathematics and education in general. But as part of my talk, I will be given use cases, specific use cases in mathematics teaching, and also discuss the implications of how the advent of this artificial intelligence has disrupted the normal way of delivering the mathematics curriculum. So to start my presentation, I would like to locate the recent developments in the parts of history so that people are aware of what is going on. So throughout history, there have been these technologies that can be pinpointed to have changed the course of development, of human development. And they then form part of what is known as industrial revolution. Like in the second industrial revolution, you had electricity, which was the main technology which came in, and then it then transformed a lot of operations, and it changed the way we do business, the change we teach, the way we teach, and the way we live as society. So through the advent of electricity, you had other technologies that came in, like telephones, also automobiles, aeroplanes, and so forth. So in the third industrial revolution, you had the internet, which is what then gave rise to other developments, like telecommunications. A lot of devices use the internet. Now you have artificial intelligence, which has come to the stage of maturation. So it has evolved to the point that it can now be applied in a wide range of tools that we use on a daily basis. So now, along with the fourth industrial revolution, in the education sector, there's a concept known as Education 4.0, which basically takes note of the integration of technology, in particular artificial intelligence and associated technologies now in education. So we are now aligning these new technologies in education along with the fourth industrial revolution. So then what tends to happen is that it causes a shift in the way we do traditional teaching and learning, because it calls for flexibility. It calls for teaching and learning to focus more on the student and less on the teacher, in a concept known as student-centered model. So then the attempt is to make sure that students are at the center, and as we teach, we are preparing them for a technology-driven workplace. So on the left of this slide are several aspects of what this Education 4.0 is. So it emphasizes on skills, it emphasizes on collaboration, it emphasizes on problem solving. But more importantly, which is what I will focus on in this presentation, is how it enables personalized learning. So I'll be giving examples of personalized learning as a specific focus of the presentation. So what is the artificial intelligence that we are talking about? First, you must note that artificial intelligence is not new. It has been there for a long time, for close to 60 years, because the concept or even the name artificial intelligence was coined somewhere in the 1950s. So since then, there have been different types of artificial intelligence techniques which have different applications, like machine learning, neural networks, deep learning, and so forth. So what is really transforming the way we do business is a generative type of artificial intelligence, which is the small circle at the bottom here. So this one focuses on creating content. So it seeks to just generate new content based on the way it perceives information. So the excitement then is about the democratization part of artificial intelligence, which basically means that the artificial intelligence is now at the point where it can be spread out and put in the hands of almost everyone to apply in many use cases. So that is the concept of democratization. In this slide, I just give a visual illustration of what we mean by democratization. If you look back in history, you can equate the democratization of artificial intelligence as the transition from typewriters to personal computers. In the past, you had maybe one or two people who are skilled at typewriting, but now when the technology transformed, it then gave birth to personal computers, which had more functionality in the sense that they came with software. The software could be used for more functions, to solve more problems. So democratization then often leads to increased accessibility, of a particular technology. So when you are talking to AI, democratization would mean that the AI is now available to all of us to use. You only have to access it through a browser, just like you access Google, and you interact with it through simple English language commands. So it leads to, there are some efficiencies, just like moving from a desktop to, a typewriter to a computer. The personal computer boosts efficiencies, and for us in education, you can agree with me that thanks to the availability of computers, you are now able to innovate, you are now able to solve your research problems by using software, and the quality of the product, the research output, improves because of the availability of the software. So the same will apply with this new technology called generative AI. You can use it to improve your research. You can use it to improve your productivity. I highlighted that I want to focus on personalized learning. So what is this personalized learning? Personalized learning refers to tailoring education to make sure that it meets the individual needs of a student. So you use technology to empower a student so that the student is in control. So there are many aspects of personalization that are facilitated by artificial intelligence. I will discuss some of them as we go along. So why is personalization important in education? So there is this image here, which is a classic image that shows the difference between equality and equity. What we want to achieve in education is equity. If you look carefully on the image on the left, which illustrates equality, equality means you treat everyone the same. You assume that it's a one-size-fits-all. You give them the same treatment, the same resources, and don't account for individual differences. So you'll see that from the diagram, somebody who is short has no chance of participating, has no chance of succeeding. But when it comes to equity, equity means you adapt things. You make sure that everyone gets the support that they need. You customize the support to give those who are maybe disadvantaged an opportunity to succeed. So artificial intelligence enables the personalization in many ways through adapting explanations, offering flexibility, and so forth. So I'm going to give examples of what we mean by that. So these are some of the ways in which the artificial intelligence enables personalized learning. It offers adaptive approach. So that means that if you are using an artificial intelligence tool, you are able to adjust the responses to maybe vary based on how the user, in this case, meaning a student, how the user engages with the tool. So the tool would be able to detect that, oh, okay, this person needs help here, and adjust the way it interacts or explain things. So it can clarify explanations based on, the student's needs. And you can also generate customized problems based on the student's needs and the areas of difficulty. But more importantly, it has the ability to offer interactive learning where students are encouraged to dialogue, to have a conversation with the tool and ask questions, seek clarifications. So this is what is known as active learning. Instead of static learning like in a book where the book has one fixed explanation, here you can vary the explanation. If you don't like the explanation that has been offered, you can request for an alternative version or a simplified version until you understand what is being taught. So interactivity is very important when it comes to personalized learning because it can allow for stimulating other sensors in terms of what the students need to be able to understand content. Like if the student understands using multimedia, that would be made possible. And there's also, I've spoken about the adaptive learning where you adjust the level of difficulty based on how the student performs. And also from the teacher's point of view, you are able to adjust or set customized assessments based on the learner's level. There are other important things that can be facilitated by artificial intelligence, like providing instant responses, like when a student is studying and wants to understand a particular concept, you are able to get an instant response instead of waiting for an instructor or a tutor or assistant who may not be available to assist you at that point. So can use this as as a support tool for getting immediate help. So let me just give some practical examples of the interactive dialogue. So here is an example of how you engage with one of the AI tools. So here you have a student who's struggling with solving, a mathematics problem. So you engage in a conversation, you inform the tool exactly what you want, you inform the tool how you've approached the problem, you inform the tool where you are struggling, and you request for assistance. So as if you are talking to a person. So this is one of the recommended ways of using these tools. So this is a typical response. The response is based on, you will note that the response is itemized, 1, 2, 3, 4. Each item responds to the specific request from the instruction that you gave. So it takes you through step by step, and once it's giving you the solution. So this is like, instead of just giving you the answer, it will be attempting to teach you, to show you how things work. Then in the end, at the bottom, you'll see that the tool will invite you to indicate if you still need further clarification. So this is an attempt to engage you further in a conversation until you understand what it is that you are asking for, until your query is resolved. But now, how can we trust that these results are accurate? I think you may have been aware that when these tools were first popularized, early in 2023, late 2022, those models that came in early had a lot of inaccuracies, especially when it came to mathematics. So they were giving incorrect results when, if you are not careful, you would take them to be a fact, and then obviously that was doing a lot of damage to the education sector. So since then, there's been constant measuring of the performance. So, and also when the new models are being introduced, there's an attempt to ensure that they give accurate results, they improve accuracy. There's this phenomenon known as hallucination, which basically means giving inaccurate information and presenting it as fact, whereas it is false. So the newer models would have less hallucination. So I wanted to draw your attention to one type of test which they use to measure the performance of these AI tools. So there's this one called multitask language understanding, massive multitask language understanding. So what basically that means is you have a collection of thousands of questions taken from 57 subjects. The subjects are arranged including maths, physics, computer science, philosophy, history, 57 of them with thousands of questions. Then the AI tool would be required to attempt these questions. And so for humans, if a human being, an average human being who is not an expert, were to attempt to answer the question, the human being would get around 34.5%. Whereas an subject matter expert would be expected to get 90%. But note that this subject matter, you can only get 90% in a component where you are an expert. Like you can get 97% only in 90% only in mathematics if you are a mathematics expert out of the 57. So this is one of the tests. There are other tests that have been used. There is this body, research group at Stanford University that keeps track of the comparison between the artificial intelligence and humans in terms of problem solving. So from this graph, you can see that in issues like image classification, the AI has long passed human baseline, like somewhere around 2014. Then there are other tests. The multitask language understanding is the second line from the right. Then I want you to pay attention to the last line in purple, which is mathematics, performance in mathematics. You will note that as of 2023, the performance there in mathematics was less than the human baseline as of 2023. So let's see what has happened. In 2024, we started seeing reports that for the first time you have some artificial intelligence tools that are starting to perform better than experts or better than smart experts in an area. Like the first report came in on the 25th of July, where one of the AI tools from Google participated in a mathematics Olympiad and got silver medal, which is quite rare even for human beings. So we're starting to see rapid improvement in the performance when it comes to mathematics. Then most recently, if we fast forward to this week, so what we saw this week was a release of new AI models, which were released by a company called OpenAI. OpenAI is the company that is responsible for manufacturing one of the popular chatbots known as ChatGPT. So what you can see from this graph, so in this measure, you have different AI tools that are measured for IQ. So what you'll notice that all of them are less than 100 in terms of performance because they are, I would say they perform less than the average human being in terms of IQ. But as of this week, we started seeing an AI model that is now over the curve. So it has an average IQ of 120, meaning that it's way above average. So 120 in terms of IQ would be better than 90% of the human population. So if you're interested in reading about this test, you can go to trackingai.org. So they have more of the details about that. Then also, it's not just about IQ. So these results were published by the OpenAI, the company, just to illustrate what is different between these new models and the old models. So what they highlighted is that these new models can now think, they can now reason just like a human being. So that is the difference. Whereas the old models could not really think and reason, they were simply retrieving information that they've been trained on and predicting the next word. So what you saw as an output was simply an estimation or a prediction. The difference is that the new AI models can now reason, can now think, in addition to being able to retrieve information that they've learned. So these are some of the benchmarks. So I want to draw your attention to, let's say, the bottom left. If you can look at college mathematics, the performance in college mathematics was 98%, formal logic 97%. Then also, more interestingly, even on PhD level science questions, the performance is quite, astounding, because like on physics, for example, PhD quality, PhD questions, the performance is like 92.6, 92.8. So that is what we are now starting to see in terms of improvement or advancement in the capabilities. So there's this other benchmark which now compares the performance of the new versions of the AI that were released this week against the standard versions. So with this benchmark, they are comparing, on the x-axis, you have a range of these AI tools. You have Gemini, you have the old ChatGPT, you have Llama, you have Cloud, you have Mistral, all these tools. Then you can say that they are really close to each other. But as of this week, you have this new one, which is now off the charts. It beats them by a wide margin in terms of performance in mathematics. So this is what we as mathematicians, we should be paying attention to. There is this other benchmark that was released comparing the performance in competition level mathematics, like the International Mathematics Olympiad. So here they're comparing the old version of their model against the new. So you'll see that if you compare the green with the orange or what's this other color, or pink, you'll see that the performance, the improvement is at least 5 times, sometimes more than 6 times. So in the middle here, this is the improvement in coding. So the coding capacity has significantly improved. And on the far right, that's a comparison now between PhD level science questions and the AI tools. So you'll see that an expert PhD would score 69.7, whereas these tools are scoring higher, 78 and 78.3, respectively. So what does this mean for us as teachers? So the first thing that you must note is that there must be a shift or a change in the way you give assessment, in particular homework. So what I've given you here are two examples of this shift. In a course in numerical analysis, so on the left, like 2 years ago, the left would be a typical assignment or homework where the student would be asked to create a MATLAB code that solves the Newton's method. The Newton's method is one of the methods that we teach in typically third year mathematics. So that would be a homework exercise, but you can't give such a homework now because you have these AI tools that can do the job. So we didn't know if it's done by the student or the AI tool. So how I've adapted my assessment is instead of asking the students to create just a MATLAB code, I ask them to create a product, to create an app that applies the Newton's method. Now, they do not have to be able to code. They do not have to be computer science students. These are mathematics students with no background in coding. But they can use these AI tools to create apps because they allow you to input instructions and request any product, generate anything, whether it is a solution in the form of text, a solution in the form of an image, or a solution in the form of an interactive app. So this is an illustration of the shift that I have had to implement in the way that I give assessment. So this would be a typical output. So after giving that instruction, after a few seconds, you get a complete app which the student can use. They can input their own functions, play around by varying the number of iterations and initial guesses, and clicking the button, you get the result at the bottom. So for students, then they would have to then manually check using, a manual calculation implementation of the method to then evaluate the accuracy of the AI. So the task shifts from generating code to now generating an app which to learn or to validate the output, you must then apply the techniques that you have been taught in class to solve. So this is just one example. Another example, in this example, the output was a table, the instruction was give a table. So in this example, if you vary the instruction, you can instruct it instead of giving you a table, you instruct it to give you the text, to give you the step-by-step solution. So it can easily do that. It does that. So here on the right, you can see that it is giving the steps for the Newton's method. So this is useful for students because they can then go through the steps just to assess if this thing gave the correct result. So it's not just limited, there's a lot you can do. Here, this is another example which I gave as homework. So here I asked them to generate a simulation now for a different, this is a problem in operations research, and I specifically asked them to ensure that the app that they generate is able to check and have quiz-like elements. So you see here, the students can then play around with it, enter their own values for transportation problems, and then solve, check, solution. And if the solution is right, that's what you'll get on the right of the screen. If it's wrong, then it will inform you that your answer is wrong, so you try again. So this is now a gamification element, which I think, it's a fun way of learning for students. So in addition to using the AI to give them an instrument which they can use to test and validate if their solutions are correct, it can be a fun way of learning. Of course, what are then the challenges and ethical considerations? So one of the challenges for us as teachers that this technology is developing very fast, and it's not giving us enough time to pause, reflect, and also learn to get comfortable with it. So the rate at which it is developing is just way too fast for us as teachers to cope. And also, because of the changes and the capabilities that it is coming with, particularly at research level, where you now have an AI tool that can practically, if you give it your research statement or your proposal, research proposal, it can practically work through the rest of your PhD if you are able to prompt it correctly. So then this then calls into question if the way we offer our research, problems has got to be changed. So we need to adapt our curricula to take into account the fact that our students now have access to these tools that have got advanced capabilities. And also in terms of the assessment methods, there may be, particularly when you are giving your students offline assessments, like take-home assessments or assignments which they have to do overnight or have to bring back after several days. You must note that they now have access to tools that they can use to solve. So it calls for responsible use to avoid these academic integrity concerns, where we as teachers, we have to teach these students how to use these tools responsibly. And yeah. So what does the future of learning look like? Well, one thing that will change, first of all, is that the focus will now shift from delivery. You will not go to class to say to deliver content, to teach them the Newton's method, for example. So students would be expected to learn on their own, and then the classroom interaction will be more of a facilitation instead of just dishing out information. So obviously education will now require additional skills, like AI literacy. We need to emphasize on critical thinking, because if the students rely on this technology without applying themselves, then they will not benefit from the education, because their critical skills will not develop. Then the last point is that it is now clear that the technology will disrupt the way we deliver our teaching and learning process. Yeah, so I guess with that, let me thank you once again for giving me the opportunity. I have, I took about maybe 35 minutes or so, just to allow for questions if there are any. So I end my presentation by indicating that you must not think of AI as a tool or a software. It is just a strategic direction. And also note that the AI is not out here to compete with you or to replace you or to prove that it can do better than you. It is here to assist you. So to benefit from it, you can use it as an assistant. Think of it as a tool that is out to assist, not to compete with you. So thank you very much.
Thank you, Professor Mutsa. I think with what I've heard, this is really something interesting, leaving us with some questions, of course, in terms of how we go about our business in classroom, research. So there's a lot to think about. So I also like the way you concluded. So we are now going to open this platform for questions. We've got the microphones. They will be passed around. Let's start with that. Can we see if we've got people who would like to ask questions, even if it's comments, that is welcome. Let us start. You can raise your hand. Okay, let's see those who have questions first. Thank you.
Thank you, Professor Mosa, for a wonderful presentation. I just want to check with you with respect to regulation of this AI, especially within the institutions. And as you have rightly mentioned that, of course, we need to make use of it responsibly. And some of us are aging, and our students, they may be technologically advanced. So I just want to check with respect to the supervision, especially of postgraduate students. How can we really make sure, because sometimes you give some tasks to students to go and work on, and for some reason they may go and make use of AI and just take things from AI and present it to me, and when I look at it, I will say, yeah, this is a good work. So I just want to check with respect to the regulation of AI. And the other point that I want to check is the aspect of critical thinking, whether this won't also be compromised along the way. Thank you.
I'm not sure if I should respond now or wait for more questions, but while I wait for direction, I can just quickly respond to these two. The first point on the regulation within the institution, that must be a priority. It must be at the top of your mind, because if you do not have, there seems to be an echo. I don't know if you can hear me clearly there.
Yes, we can hear you clearly. You can
Okay. Let me proceed. So, with regulations, it is a priority that institutions must develop their own regulations for responsible use of AI, just to guide the students, to guide the instructors, to guide the supervisors. When it comes to research, for example, there are elements that you can call them acceptable use, like mechanical things, like of cleaning up a draft that a student has written, to make sure that the ideas are presented in a logical manner and so forth. There is clarity, proofreading, editing. That is acceptable use. However, it is a bit of a gray area, especially after this week, where we saw the AI now advancing to the point where when you give it a project proposal, even for mathematics, it will be able to do the analysis. I can tell you that since they released the model on the 12th of September, just this week, I've spent every day just testing how well it performs in the problems that I've done with my students, the problems that I've done as research problems. It is quite scary that it is able to solve some of those problems. You give it an equation, then you instruct it to solve that equation using a particular method and generate the code. What we would consider then to be critical thinking, generating the code, the stability analysis, the consistency check, the rigorous mathematical proof, is no longer critical thinking because this tool can do it in less than a minute. So there is a lot of confusion. So it calls for us as practitioners to sit down and really look at how we can react to this advancement, because they are a reality. It's too early to say because the new models were released just this week. So we haven't had enough time to even understand the limit of their capabilities. But it is a cause for concern. For your students who are at the stage of maybe writing a proposal, they need to really reflect carefully on what they think is an innovative research question. Because if that research question can just be solved, can just be, if the project can be designed and all the way to a result section in less than a minute or two, if you're able to prompt it correctly, then it raises concerns. I can't say I have the answers. I used to think I have the answers because I knew the limitations of the previous models of AI. But now they've advanced to the point that we don't know where it will end. But they keep improving and improving.
Okay, thank you. Prof. Motsa, you were asking if you could listen to all the questions and then maybe answer after that. I suggest we continue this way. So,
okay.
Yeah, one at a time. So we are continuing. Thank you.
Thank you.
Do we have any hands?
Am I audible? Okay, thank you very much. Am I audible? Okay, thank you very much, Prof, for the insightful presentation. I was just wondering also as a student, as you have mentioned that AI tools are continuing to advance. So I just wanted to know, what are some of the skills that you would recommend for us students to have or to gain in order to stay competitive in the job market? Thank you.
Prof, did you hear that?
No, please repeat the question for me.
Could you
Thank
please,
you very much, Prof.
Sorry?
Can you hear me now?
Could you please come closer?
Am I audible now?
Yes. Just here.
Here. Oh yeah, now I think it's fine. Can you hear me now, Professor?
Yes, I can hear now.
Okay, thank you very much. I was asking, from the students, now, as you've mentioned that some AI tools even have IQ of 120. So we're wondering, what skills should we now prioritize as students so that we can stay competitive in the market industry? What are some of the skills that you would advise us to acquire so that we stay competitive? Thank you.
Did you hear that, Prof?
Yes, I got it. If I got it correctly, the question was from a student's perspective. Given that the AI has now advanced with IQ, which is now up way above average, what are the competitive students? Would they be competitive? What sort of skills would I advise them to focus on? Let me repeat what I said in the conclusion. that the idea for AI is not to try to compete with it. It's not to try to demonstrate that you have a better IQ or you can do the job better than an AI, because you cannot. You cannot do that. The AI, the design of an AI is to help you perform tasks more efficiently and more accurately. So they will always improve. This model is only 120. The next one will be even higher than that. This one is at PhD level in terms of understanding. The next one will be even higher than that, which means the aim is for superhuman. So in terms of skill, as a student, you must learn to collaborate with the AI. You must learn to use the AI to solve problems. So I gave an example of how I'm assisting my students in that regard. I teach them AI skills so that they are able to develop products, because when it comes to the workplace, the interest there is on solving problems or developing products, not on how smart you are. In fact, the smartest student who has an IQ of 200 is not very useful in the workplace, because the workplace demands certain things, demands delivery. So you must be able to learn AI literacy, be comfortable with the AI, be comfortable to using AI as a support tool, just like you are comfortable with software. You are comfortable using a calculator. You are comfortable using any other technological device. So AI is not different from software. So the only difference is that it can solve a wide range of tools, and it is now based on not just mechanical things, it is based on reasoning, it is based on thinking capacity. So as a student, my advice to you would be to embrace the AI, learn how to use it responsibly in your education and in solving tasks. Use it as a tool, as a support tool. Don't try to compete with it, such that when you're asked in an interview, if somebody asks you, why should you be hired if AI can do this? Your response should not be, I can do it better than AI. Your response should be, I use AI to perform this task. I can do more with AI to perform tasks. So don't compete, collaborate with it.
Okay, thank you, Prof. Just one aspect to your attention, delegates. So this call will be ending in 6 minutes, and we will now want to move to a comment by the Executive Dean. And Prof. Motza, if we are cut out, we will apologize for that, and we thank you. Then the rest will come via the email. We are sorry about that. So let's hear the Executive Dean. He has a comment. Prof, you can go.
Thank you very much, Chairperson. Sandile, good afternoon. Good afternoon, Sandile.
Prof. Motza?
Are you still there?
Yes, I can hear, yes.
Thank you very much, Sandile. I want to make 2 comments. The first one is to thank you and the organizers that you come and give one of these plenary talks. We really thank you for that, and also congratulate you on your appointment as the head of the faculty. So the first thing that I want to say is that given the things you are talking about, I was wondering whether you are aware of the Natural Science Deans Forum in South Africa. And I'll tell you why I want you to know about it. Because the things you are talking about, for me, they should feed into policy in the way in which we want to teach not only the sciences, but in the way in which we want to teach in general. So it will be something that everybody will be interested in, nationally and in SADC. So there is a meeting of the National Science Deans Forum. So deans in Swaziland, deans in Botswana, deans in Zimbabwe, deans in Namibia get invited to that. I don't know what's the situation after COVID, but before COVID, everybody was invited. So I'm going to get the email from the organizers and forward you that email so that if you have funds, you can join your colleagues in Cape Town towards the end of October and talk about these things. Is he out of the line now?
Prof. Motsa.
Are you there, Sandile?
Hello?
Can you hear me, Sandile? Sandile?
Hello? Yeah, I can hear now. I was not hearing
okay,
before.
okay. I'm talking about the National Science Deans Forum. I'll send you details to that through the organizers, so that if you are able, you attend the meeting in Cape Town, where you can advance these ideas. That's the first comment. The second one, which is important for this faculty, is that now that we have you here as the head of the faculty, I think we should now seriously speak about collaborations between the Eswatini University and the Faculty of Science and Agriculture in two respects. One, it's sharing the syllabuses, as you've already indicated, okay? And then two, from next year, our doctoral students will be required to defend their thesis. So if we have a collaboration, then we have a baseline from which to start, so that if we are required to ask for external, international examiners, we are able to just walk across and say, okay, Professor Swandile in chemistry, who is going to assist us? Then you tell us that. If we have that collaboration, you will have mobility for staff and students, right? Forget about xenophobia. We are not xenophobic at the University of Limpopo. So we want students from your faculty, if we have a collaboration, to come and spend time at the University of Limpopo, especially around this time of the year, when we have this gathering where these youngsters come together and talk about what they do. That's what I want you and I to pursue. Thank you very much.
Prof. Matsa, are you still there?
Yes.
We've got him. It's done. Okay, thank you, Prof. Siwaya, for the comments. We'll follow up via other platforms. And I think what remains for me is to thank these delegates. Thank you so much.
letterhead.
Good afternoon, everyone. My name is Masibu Katsepo, and then I'll be presenting a paper on environmental and animal factors affecting milk production of Toggenburg goats in South Africa. Toggenburg dairy goats it is initially bred from Switzerland. It is a very much crucial to dairy industry due to its hardiness, adaptability, and high milk yield, and as well as the fat content. So this goats, optimization of this goat requires us to have an advanced knowledge and understanding on the several factors between animal factors such as temperity, kid sex, litter size, and as well as the environmental factors such as birth year, kidding season, and as well as the agroecological zone. So they have, their milk contributes significantly to the dairy industry because of its desirably high butterfat and content of the protein. So these goats have got the potential to produce a higher milk, but it all depends on the several factors involved. Thus, their performance largely is influenced by both environmental and animal factors. The figure one there is just showing the Toggenburg goat. With the increasing significance of Toggenburg goats, South African dairy industry still produces low milk from these goats, and this is because South Africa experiences the extreme climatic conditions which affect the productivity of the goats, as well as their reproductive capacity, and as well and the overall productivity. So the dairy industry is very much crucial in providing the milk protein to the South African industry to do away with the issues such as the malnutrition or poverty. Understanding environmental and animal factors is very much essential for goat milk productivity and sustainability, as well as the maximization of the milk production. So according to this author, there are several studies on management and nutrition influencing goat milk production. However, according to our best of knowledge, there is a limited literature on impact of environmental and animal factors on South African Toggenburg goat milk yield and quality, which necessitates the need to carry out this study. The aim of the study was to evaluate the influence of environmental and animal factors on milk production of Toggenburg goats in South Africa. The objective of this study was to assess the impact of environmental and animal factors affecting milk production traits of Toggenburg goats in South Africa, and then the hypothesis is in line with the objective. The study utilized the South African agroecological zones such as arid, semi-arid, subhumid, and temperate zones. The study used the records of 2,417 Toggenburg goats which were born between 1990 and 2018. Purposive sampling and convenient sampling were followed to select the zones that had the Toggenburg dairy goat, and as well as to select the Toggenburg dose with the necessary milk data, respectively. Conventional standard for raising Toggenburg goat was followed. This include breeding, vaccination, healthcare, housing, among others. The study design was followed using the longitudinal observational study design. Secondary data was collected from the logistic database. The preliminary and advanced data editing was done using Microsoft Excel and the Minitab software, respectively. The data analysis was analyzed. The data was analyzed using the ANOVA, which were fitted in the general linear model in the Minitab. Fisher's LSD was used for mean separations. On the results, Table 1 represents the effect of kidding season on milk production throws of Toggenburg goats, as we can see on the highlighted green boxes there. The result shows that summer and autumn performed better in terms of the milk persistency or the maintenance of milk after the peak, and as well as the milk fat content, and as well as the milk index. So this current study results are aligned with the reports of this author and this author, 2024 and 2021, that revealed that certain traits like persistency or fat production are mainly optimized in summer seasons and autumn seasons due to favorable nutritional and environmental conditions. The figures represent the effect of birth year, which shows how the milk traits were behaving amongst the year from 1990 to 2017. So the results that we got represent or shows that the goats that were born from 2014 had high capabilities of producing milk and the content. So this study says milk production variations are influenced by environmental and genetic factors, with a peak which was seen in 2014, possibly due to selective breeding or better management. So this author also observed the same results. Table 2 represents the effect of agroecological zones on milk production traits of Toggenburg goats. The results highlight that subhumid zone and temperate zone performed better in terms of milk index or lactation index. So this means that a high milk yield or to maintain milk lactation index are mostly achieved in these two zones, arid and temperate zone. The study reveals the same results by this author, with subhumid and temperate zone experiencing higher indices, milk indices, while arid and semi-arid environments negatively impact the lactation due to high temperatures and low rainfall. Table 3 represents the effect of dam parity, which is one of the animal factors, on milk production traits. This is the mistake, dam parity on lactation, milk yield production. Yeah, it's a mistake there. This table represents the effect of dam parity on milk production traits. So generally yielding, this result generally showing that parity number 3 generally have got higher yields. So the results correspond with the result of this author, who documented that goat milk and component yields increases with parity until parity number 3 or 4. Then it declines due to aging or lactation stresses of the dam. Table 4 represents the effect of kid sex on milk production traits of Toggenburg goats. So the results showed that kid sex had no significant effect on the production of milk, meaning that the sex of the kid has nothing to do with the production of the milk. Therefore, this study confirms that the environmental, dietary, and genetic factors, rather than the sex of the offspring, significantly influence milk production traits in Toggenburg. This author reported the same results. Table 4 represents the effect of litter size, which is also one of the factors, the animal factors affecting milk production traits. So as per the tables, the results showed that litter size 1 or single birth better improved in milk persistency, and then the triplet birth achieved higher yields in milk production. So these results are similar to this author, which indicated that multiple births in goats accelerate the decline in milk production after the peak, which in this study it is seen on the parity number 1 on the milk persistency there. Despite their steady milk despite their steady milk supply throughout the lactation. In conclusion, the study concluded that despite non-significant effect of kid sex, kidding season agroecological zone, birth year, dam parity, and litter size significantly affect milk production and its components in Togenbe goats. It is therefore recommended that the goat farmers and breeders are advised to adjust their management strategies to optimize the optimal conditions for growing Togenbe goats. Future studies should investigate more on factors affecting productivity of goats for more validation of the current study. So these are some of the references that I've used. I would like to thank the university, the faculty, my supervisors, Professor Tada, Professor Tias, and the research, Tias research team, the food BEFCITA, and our department. I thank you.
Thank you, Mr. Machebuka. I am now opening the floor for questions. Do we have questions or comments? One. Two. Okay, we have 3 minutes for questions. Just give him time to answer.
All right, thanks for that presentation. I just wanted to find out in terms of your analysis, I think you established that there are certain regions where the milk production, agroecological regions where the goats in those regions produce higher quality, quantity, maybe quality in terms of the milk. Now, when you started analyzing other parameters, did you separate the goats based on the zones, or you were analyzing them all at once?
The goats were analyzed. Firstly, the goats had got the regions. That shows that they were from this region. These goats, so they were analyzed all in all based on those regions in the software.
Yeah, but I think when I look at the results, I mean, correct me if I'm wrong, it looks like you were then analyzing everything at once. You didn't split in terms of the regions to say the regions from this sub-ecological zone are not impacted by environmental whatever when it comes to A, B, C, D, E. That's why I'm asking the question.
All right, thank you.
You can quickly go to your results.
From where?
From the presentation.
Sorry.
I mean, where they're talking about the regions and zones.
Oh, this table. No, this one is the effect of dam parity. Effect of kid sex. Which region? This table represents the effect of kid sex. The tables that has got the regions are these ones. Yeah. Okay. Yeah, I understand.
Okay, thank you for your presentation. My question, maybe I missed it, which genetic factors did you analyze to check if they affect your milk production? And the second one is just a comment. When your results, you were not discussing them, you were just telling us your results and then telling us that they agree with someone else. So you are not telling us what contributed to your results, but please answer the genetic factors that you used.
The genetic factors that I've used?
Yes, you said you checked the effect of genetic factors on the milk production? Which genetic factors?
These are the not genetic factors, animal factors.
You don't have genetic factors here?
No.
The student is working on environmental and animal factors. The last question, 1 minute remaining. Okay, in the absence of questions, can we please clap hands for him? Okay, there are changes. We're going to allow only one question per researcher. So without any waste of time, ladies and gentlemen, allow me to call our master student, Katlego Mabale, and the student will be presenting an evaluation of the impacts of urbanization on Klip River wetland degradation in Joburg, South Africa. Can we please clap for her? Oh, sorry. I thought you were here. Sorry.
No, no. That's him. That's him. Good afternoon, ladies and gentlemen. I am Katlego Mabale, a SACNAS pre-registered water resource scientist, signifying that I'm recognized for assessing this paramount resource. However, I am optimistic about taking you through my study, which is titled An Evaluation of the Impacts of Urbanization on Klip River Wetland Degradation in Johannesburg South, supervised by Dr. R.T. Akambi, co-supervised by Prof. J.M. Letwalo, may his soul rest in peace, and lastly, co-supervised by Ms. M.G. Muntwiri. This is the outline of my research. Urbanization. Globally, urbanization has impacts on various aspects of human life, including natural resource, economic development, and also human habitat. In the 21st century, wetlands, which are known as the transitional zone between aquatic and terrestrial ecosystem, are prone to degradation due to anthropogenic activities. This is a major concern because wetlands offers significant, important ecological services such as water supply, cultural and recreational services. Johannesburg South in South Africa's Gauteng province, it's a hub for economic growth, and there's inward migration as people look for better opportunities. Particularly in Johannesburg South, the gold mining industry in the area has led to the contamination of the Klip River wetland, as there's reports about the accumulation of acid mine drainage. One may ask, what's acid mine drainage? Acid mine drainage basically result from when sulfide minerals such as pyrite, when they are exposed to water and air, they go through oxidation reaction, resulting in acidic wastewater effluents which are rich in heavy metals. Overloaded wastewater treatment plant in the area are discharging effluents that don't meet the required standards into the Klip River wetland, putting pressure on the quality of the water. In this case, nitrates were reported to be in this wastewater. Consequently, this will cause eutrophication issues at Vaal River. The incentives behind the selection of this study is because most studies around the Klip River wetland have only assessed the quality of the water. So this study intends to link water quality and also socioeconomic dynamics of people living around the outskirts of the Klip River wetland. So I also noted that the Klip River wetland supplies the bulk water to Vaal River, which is the lifeline of most areas in Gauteng. And also I looked at cultural and recreational services that the wetland offers. The aim of the study is to evaluate the impacts of urbanization on Klip River wetland degradation in the Johannesburg South through water quality assessment, and also the community perceptions and attitudes on how urbanization has impacted their livelihood, and also to evaluate factors related to urbanization in these communities which are known to interact with the quality of the water. Listed here are the research questions. The Klip River wetland is a wetland of about 100 kilometers. It flows from Rodebord to Val River. It consists of 3 subcatchments, the lower, middle, and upper, middle, and lower subcatchments. But then this study focused on the upper catchment because the upper catchment is characterized by Klipsprout, which is a tributary of the Klip River wetland, and also there's Soweto settlements, Lenasia, and there's mining industries in this area. And shown there is the geographic location of the Klip River. For addressing objective 1, 4 sites were selected along the Klip River wetland, mainly focusing on potential source of contamination, and also the samples were collected seasonally because previous studies have reported that seasons have drastic impact on the quality of the water, and also the collected samples were analyzed for 18 water quality parameters. But before then, there was preservation methods in terms of cooling the samples and transporting them into the laboratory, analyzing them within the required time to avoid contamination. Water samples were analyzed for multi-parameter, turbidity meter, were used to analyze. These are actual instruments that were used to analyze the samples, including multi-parameter, turbidity meter, the spectrophotometer for physicochemical parameters. Then ICP OES was used for chemical parameters that included anions and cations and heavy metals. So for biological parameters, I don't know what happened on this slide, but underneath there's how I did my biological parameters. I used the correlate 18 technique for enumeration of E. coli and total coliforms. Depicted here is the analytical instruments that I used to analyze my samples. A water quality index was used to determine the overall pollution of the Klip River wetland using the listed equations. So this was the water quality class that was used to rank the overall water quality. And the multivariative statistical analysis were used. Origin Lab was the statistical tool used to assess Pearson's correlation and the principal component. Principal component was used to assess the source of pollutants, and the Pearson's correlation was used to address the relationship between the physicochemical parameters and the biological parameters as well. So for addressing objective 2, which is community perceptions and attitudes on how urbanization has or impacts the local communities, a questionnaire tool was used for this one, whereby Protead land, which is on the southwest of Soweto, was selected, and also Lenasia. These 2 areas are in the vicinity of the Klip River wetland. So a sample size of 192 respondents was determined using a Rasoft calculator, whereby both total population of Lenasia and Protead land were used. So this study targeted respondents who are 30 years and above and have lived around Protead land or Lenasia for 15 years and above. So there was 2 sections in this questionnaire. There was closed-ended questions and open-ended questions. For closed-ended questions, we asked socioeconomic background such as age and their educational level, because it is known that some of these factors can be vital in addressing the quality. So open-ended questions included land use types, climate change, solid waste management, drainage systems, and wastewater treatment effluents in the locality of the Klip River wetland. For this objective, a combination of assessment tools to address the factors related to urbanization were used, which is objective 1, mainly focusing on the part of open-ended questions, whereby most factors were able to were analyzed actually. So this thing will also be augmented by the objective 1, mainly on the principal component analysis, because it is used to assess the source of contaminant, and also secondary data will be used. These assessment tools will be used to drop conclusions. So this table shows the selected parameters which surpass the guidelines. It can be seen that turbidity, which is the cloudiness or the cloudiness or the clearness of water, surpass the South African water quality guidelines for domestic use. So comparing both seasons, it can be stated that dry season recorded lower values than wet season.
This is mainly because sediments are washed on the ground into the river during rainy seasons. And this also means that because all the seasons surpass the guidelines, meaning that both water collected along the seasons was not clear, it was kind of cloudy. So for COD, which is the amount of chemical oxygen required to break down organic matter, surpassed the guidelines. Comparing two seasons, we can see that wet season recorded high values, meaning that wastewater that also gets free movement was also one of the contributors to the COD. But looking at manganese, which is a metal, dry season recorded high. This was due to, or maybe it might be attributed to evaporation, because now urbanization, it's not a process that occurs instantly. It's a long-time process. I tried to take source the historical water quality data from 2011 to 2023 from about the Klip River from the Department of Water and Sanitation, and also try to compare that with what I got. So Rand Water Board recorded high manganese as compared to what I found. This shows that the wetland is deteriorating. Pearson's correlation showed turbidity and EC and chlorides, meaning that they bothly correlated. It's a positive correlation, mean when the other one increased, also the other one increased. EC and chloride means salinity. Turbidity can also be attributed to sediments, and EC is also as well due to salinity. This is a biplot conducted or assessed for wet season, showing that the three components were extracted with eigenvalues greater than 1, but then the biplot only showed 2 components, whereby PCA1 attributed to most of the variance, and it can be seen that it had high loading of E. coli, sodium, EC, turbidity, COD, chloride, meaning that these parameters are correlated and they originate from the same source. These sources can be wastewater, salinity, yeah, and on PCA2, only chromium, iron, and E. coli were correlated. For chromium and iron, we can say that they might be originating from mines or maybe weathering of rocks. E. coli also on PCA2 must accounts for wastewater. So for wet season, PC1 contributed most of the variance, whereby E. coli, sodium, turbidity, manganese, chloride, COD, and EC had high loadings.
Your time is up. Can you please wrap up?
Up. The time is up.
Yeah, yes.
Oh. However, PCA2 accounted 22.5% of the variance, whereby sulfate, pH, fluoride, copper, and E. coli contributed to this principal component. And it can be attributed that wastewater, salinity, and weathering of rocks are the sources of pollution. We calculated the water quality index. From this 4 sites here, we can say that from site 1 shows that the value ranked between 0 to 44, and then it denotes that the water is poor for both wet and dry season. But then for site 2, it also ranked between 0 to 44, but comparing both seasons, we can say that dry season had lower quality when comparing to wet season. For site 3, it recorded values which were between 45 to 64, denoting marginal or slightly poor water quality. But comparing both seasons, we can say that wet season was lower than dry season. However, site 4 is the most contaminated site because it recorded value lower than the other sites. The questionnaire tool is still ongoing. However, the preliminary data highlights that the respondents who are between 30 to 60 with a significant educational background, secondary to tertiary, perceived intensified land use types such as commercial, industrial, and residential. And also respondents in these areas perceived traffic congestions and wildfires which affected climate and resulted in heavy rainfalls around the Klip River wetland. And also these respondents, they perceived expanded septic tanks due to population increase. In conclusion, the comparison between the data that I assessed and also the historical data, we can say that there's a continuous water quality deterioration within the Klip River wetland. And the water quality index, the principal component, substantiates that seasons can affect water quality, and the pollution with regards to site follow these trends. Although urbanization has positive impacts on the social, the intensification of factors such as mining, industries, human settlements, wastewater treatment plants are affecting the quality of the water. And the indigenous knowledge assessed based on age and educational background are linked with urbanization and the environment, because old and knowledgeable respondents perceive the most factors which can be linked to the deterioration of the Klip River wetland. For recommendation, I recommend that this study actually recommends that there should be a continuous water quality assessment. There should be a time-to-time computation of wastewater mass balance as recommended, because mass balance can be used to assess the removal efficiency of certain pollutants within wastewater treatment plant reactors. Introduction to newly developed technology is vital for effective remediation of pollutants, because wastewater treatment plant which use the standard wastewater treatment technologies are not effectively treating the pollutants. For social aspects, this study recommends environmental awareness should be strengthened to minimize human-induced pollution. For ethical consideration, to safeguard the rights of human research subjects and to improve the validity of the study that is conducted, ethical clearance was requested from the TREC. I would like to acknowledge the following: my supervisors, Energy and Water SETA for funding this project, Capricorn District Municipality for laboratory space, and the University of Limpopo, and these are the references that I used.
Can you please clap for him? I'm asking one question for him.
Okay, thank you, sir. On your background information, you mentioned that the Vaal River supplies water to the areas in Gauteng, and from your result, you highlighted that manganese had high content, whatever. And then from my information that I know is that manganese is a heavy metal of which is toxic. So I want to know if this manganese have impacts on the health of the local members from the areas of Gauteng, and in reference of your study, how can your study assist in helping with those impacts? Thank you.
Due to time, I didn't get to a point where I mentioned the impacts of this manganese, but however, the concentrations indeed shows that manganese can show toxic impacts on humans, as this water, it is also used for domestic, as per the studies that are reported. So due to the fact that I only focused on assessing the quality of the water, I was not able to effectively outline the impacts of these pollutants. Yeah, and also the other one you said, what was the other question?
Only one question.
But it sounded like it's two questions.
It's fine, you can go and ask.
Okay, no, it's fine, I'll answer.
Thank you.
But yeah, there's effects linked to manganese, just that due to time. Yeah, thank you.
Thank you. Moving along, we're calling the next speaker, which is Katlego Nkgweng, a master's student who will be presenting the effect of carboxymethyl cellulose wax composite coating on post-harvest quality on tomato fruit. Is Katlego Nkgweng around? Okay, can you please encourage her? You are given 12 minutes to present, 3 for questions, please stick to time.
Greetings to you all. My name is Nkhoeng KR and I'll be presenting about the effect of carboxymethyl cellulose wax composite coating on the post-harvest quality of tomato fruit. Tomatoes are the most important vegetable crops that are grown in the world. They provide a wide range of benefits in human health, including carotenoids, lycopene, vitamins, and minerals. These nutrients, they help in the prevention of chronic diseases, such as the cardiovascular diseases and also cancer. Despite its popularity, tomato fruit faces several post-harvest challenges that affect its quality. One of the significant challenges is its susceptibility to rapid deterioration, resulting in the loss of texture, flavor, and the overall appeal. Post-harvest quality preservation of tomato fruit is important to maintain their commercial value and also extend their shelf life. One of the approaches that are used to prevent the post-harvest challenges is the use of edible coatings. One can ask, what are the edible coatings? Edible coatings are a thin layer of material that are applied on the surface of the fruit to preserve quality and further extend their shelf life. These coatings, they form a layer around the fruit, regulating the factors such as moisture loss and flavor. By doing so, they help extend the shelf life. However, the individual coatings, they come with their own set of challenges. For example, the CMC coatings, they can become brittle as they dry and cause cracks, potentially compromising the protective barriers. These cracks, they may permit moisture loss and microbial entry, further affecting the quality of the produce. On the other hand, individual coatings such as wax, example being the citrusian wax, they can hinder gas exchange due to their hydrophobic properties, and as a result, they can further impact the flavor and the respiration rate in the fruit. So the composite coating, which consists of the combination of the CMC and the wax, they can present solution to these challenges, as these composite coatings, they offer they manage the multiple aspects of preservation quality, which include the moisture retention and also the gas exchange. The aim of this study was to preserve the post-harvest quality of tomato fruit using carboxymethyl cellulose wax composite coating. The objective of this study was to determine the effect of carboxymethyl cellulose wax composite edible coating on the post-harvest quality of tomato fruit. Looking at the methodology and analytical procedures, the tomato fruit were bought from a commercial farm and transported at ambient temperature to the plant production laboratory at the University of Limpopo. The experiment was laid out in a completely randomized design, arranged in a 4x4 factorial with 3 replicates. Looking at the treatments and the procedures, upon arrival, the tomato fruit were washed to remove any debris on the fruit, and then after they were dipped in a chlorine solution to remove any fungal infections around the fruit. After that, they were dried, and after the fruit were dipped in the coating solutions, including the CMC, Citrashine wax, CMC plus Citrashine wax, and also the control. After that, they were dried and stored at ambient temperature. The following variables were determined at the 3 intervals. The collected data, it was analyzed using ANOVA, and the comparison of treatment means was performed using Duncan multiple range test at 5% level of significance. The statistical analysis, it was conducted using the GenStat statistical software. Looking at the results, the control samples exhibited, the results shows that the control samples, which were only treated with water, exhibited highest weight loss with the 0.5 CMC plus, with the 0.5% CMC wax exhibiting the lowest weight loss. So these results, they show that the CMC, the 0.5 CMC plus wax provided superior moisture retention, likely due to the enhanced barrier properties. With firmness, the results shows that the firmness decreased over the 9 days storage period, with the 0.5 CMC, with the 0.5% CMC plus wax coating exhibiting the highest firmness, followed by the 0.5 CMC alone and the 100% wax. However, the control samples only, the control samples, they were the ones that exhibited the lowest firmness levels. So this shows that the 0.5% CMC plus wax coating mostly was the most effective and was the most effective treatment that preserved firmness due to the protection against the mechanical degradation. And then with the total soluble solid, which is the TSS, the control samples show an increase, a sharp increase in the TSS in the tomato fruit, peaking at day 6, and after that they slightly decreased by day 9, and then the 0.5% CMC plus wax coating, it showed more consistent TSS levels, which slightly increased by day 9. So the 0.5% CMC and also the 100% wax also showed consistent TSS levels. However, there were slight fluctuations, but by the end of day 9, they decreased. So these results, they show that the 0.5% CMC wax coating, it appeared to slow down the ripening by regulating the metabolic activity, resulting in more consistent TSS levels and better preservation quality. With the titratable acidity, the control samples showed the steepest decline from day 0 until day 9, whereas the 0.5% CMC and the 0.5% CMC plus wax and 100% wax coating materials, they showed a gradual decrease from day 0 until day 9. So these results, they show that the coated fruit, the coating was able to effectively delay ripening by reducing respiration and the acid metabolism. With the TSS
You have
over
2 minutes left.
with the TSS over TA, the results show that the TSS over TA ratio increased during the day 9 storage. However, the coated fruit were able to enhance the flavor and also delay ripening, whereas the control samples which had the lowest TSS over TA ratio, and then the coating effectively preserved the organopenic qualities of tomatoes, unlike the control solution. In conclusion, the carboxymethyl cellulose wax edible coating effectively preserved the storage life and maintained the quality of tomato fruit with the coatings outperforming the individual, with the 0.5% CMC plus wax coating outperforming the single coatings and also the control. Therefore, this coating, the carboxymethyl cellulose wax composite coating, it is recommended as a strategy to reduce the post-harvest losses, and therefore it can be used in the tomato industry, especially by the poor resource packers and distributors. I would like to acknowledge my supervisors and the University of Limpopo. Thank you.
Thank you, Ms. Mkhweng. Do we have a question for her? Okay, this.
Thank you. Okay, since tomatoes are most widely researched fruits, and then it has been noted that tomatoes have their own natural ethylene content. Even if you harvest them while they're still green, they can still continue with their ripening process. So I want to know if when you apply this edible coating, doesn't it affect the content of ethylene? Doesn't it affect it, and this ethylene continues with its ripening process? I don't know if maybe you got the. Okay, fine. Tomatoes are known to have high ethylene content, of which helps them with the post-harvesting for faster fruit ripening. So I want to know, when you coat your fruits, the tomato fruit, with that edible coating, doesn't it affect the content of ethylene in it?
Okay, thank you for the question. When I apply the edible coatings on the tomato fruit, they slow down the ethylene production within the fruit, hence slowing down the ripening process.
Thank you. Can I have a follow-up question?
No, we are moving.
Thank you.
You'll ask outside.
Thank you.
Thank you, Mrs. Mkhweng. Can you please let our hands for her? The last speaker from me before Dr. Slopas will take over will be Kagiso Ledwaba. And Kariso will be presenting on comparative analysis of Hass type avocado, epicap color development and softening during ripening, a case of traditional Hass, Maluma Hass, and Camel Hass. Ladies and gentlemen, please. You are given 12 minutes and your time starts now.
Good afternoon, everyone. I am Ledoba Karisho, and I'll be presenting my research topic titled A Comparative Analysis of Hass-type Avocado Exocap Color Development and Softening During Ripening of 3 Avocado Cultivars. A case of Hass, Maluma Hass, and Kamen Hass. Worldwide, avocado are the most important subtropical fruit due to their higher nutrition and economic value. About 45 to 50% of avocados produced in South Africa are exported to the lucrative European markets, with Hass dominating the markets due to its creamy texture, nutty flavor, and color change from green to purple or black during ripening, therefore making it a preferred option among consumers. Among the existing Hass-type cultivars, Maluma and Kamen Hass are the newly developed cultivars which have recently gained popularity due to having a superior characteristics that distinguish them from the traditional Hass. These two cultivars are known for having a higher creamy texture, higher oil content, they yield higher and are more resilient to pathogen or disease attacks as compared to the traditional Hass. However, so during ripening, Hass avocado color development must occur at the same time with softening. However, previous studies have observed a color development desynchronization with softening in early season traditional Hass type. Therefore, this results in a poor color development at post-harvest. However, it remains unclear if the two newly cultivars exhibit the similar behavior as reported in traditional Hass. South Africa plays a major role in the export market of avocados. So farmers desire to enter the market early while prices are still high, and the marketing of Hass heavily relies on its color development. Now, the issue lies with the poor color development that was observed in early traditional Hass cultivar. Therefore, exporting these new cultivars without knowing their behavior may lead to rejections. Therefore, this study is necessary to address the potential challenges in color and softening synchronization in these newly developed cultivars. The aim of the study was to compare the post-harvest exocap color development in Hass, Maluma Hass, and Kamen Hass, and evaluate their correlation with softening. The objective was to determine if there are any significant differences in the exocap color of these 3 cultivars and evaluate their correlation with softening. Under materials and method, the 3 cultivars were harvested early from ZZ2 in Tzaneen. They were then transported to the University of Limpopo Plant Production Lab, where they were stored for 28 days at cold temperatures, then ripened at ambient temperatures. This experiment was carried out in a completely randomized design, arranged in a 3:12 factorials, where the 3 cultivars were evaluated for 12 ripening days and replicated 3 times. The quality assessed were the exocap color, which was measured using a visual color rating from 1, which indicated emerald green, to 6, that indicated black. They were also measured objectively using a chromometer. The firmness determination was measured using a penetrometer. So dry matter content was measured using an oven dry method and calculated using the highlighted formula. Lastly, the external chilling damage was assessed using a scale of 0, which indicated no chilling damage, to 3, that indicated a severe damage. The data analyzed and a Pearson correlation, which was used to assess the relationship between color and firmness, was conducted using GenStat. Under results and discussion, the study discovered that the interaction between ripening days and cultivars had a significant effect on the visual color. So during ripening, all cultivars exhibited an increase in visual color, with common hairs showing a more pronounced increase, and by day 12, common hairs was black, while maluma was purple and hairs violet. So this change in visual color was due to the accumulation of anthocyanins, which gave the fruit a darker color. So the following illustration shows how the 3 cultivars changed color during ripening. So on the objective color, the interaction between ripening days and cultivars had a significant effect on chroma and hue, while it did not on lightness. So during ripening, all cultivars exhibited a decrease in lightness, chroma, and hue, with common hairs showing a more pronounced decline that is indicated in a red color, while maluma and hairs had a gradual decline that is shown in a white color. So although these cultivars are similar in genetics, although they're genetically similar, they are different in terms of morphology, more especially a fruit size. So fruit size has an effect in the accumulation of anthocyanins. Maluma and common hairs were much more smaller in size as compared to the traditional hairs. So smaller fruit have a smaller cell size and a larger surface area, which resulted in a higher concentration of anthocyanins. In regards to firmness, the interaction between the ripening days and cultivars had a significant effect on firmness. So during ripening, all cultivars exhibited a decline in firmness, with common hairs exhibiting a more pronounced decrease, while maluma hairs maintained a higher firmness. So the rapid ripening in common hairs was due to having a higher ethylene sensitivity as compared to the other 2 cultivars. The relationship between color and firmness was observed. So in common hairs, the correlation between firmness was strongly negatively correlated with visual color and positively correlated with lightness and chroma, as compared to hairs and maluma hairs. So this indicates that firmness with visual color, lightness, and chroma were highly synchronized in common hairs as compared to hairs and maluma hairs. Dry matter content, which is indicated in a blue bar graph, did not have a significant effect among the 3 cultivars. However, common hairs had a higher percentage of dry matter content, which indicated maturity as compared to the other 2 cultivars. So chilling injury index, that is shown on a red line graph, had a significant effect on the 3 cultivars, with common hairs showing a less chilling damage as compared to hairs and maloma hairs. So the higher chilling damage that was observed in hairs and maloma hairs was due to having a higher moisture content. This is because excess water can damage the cells when exposed to lower temperatures. In conclusion, the study observed the differences in the color development and correlation with softening among these three cultivars, with common hairs showing a more significant change in visual color and objective color, indicating a more improved color change when compared to hairs and maloma hairs. Therefore, this makes common hairs a potential option for early export. However, it is not going to replace hairs. Instead, it is going to complement the market of hairs by providing an early season option. It is recommended that farmers optimize their crop management practices such as irrigation, fertilization, and pruning to enhance the accumulation of carbohydrates in hairs-type cultivars, as it serves as a key precursor for anthocyanin accumulation, therefore color development during ripening. My acknowledgement goes to my supervisors, the University of Limpopo, for allowing me to carry out the study, ZZ2 for providing me with their fruits, and FoodBev for their financial support. Thank you.
Thank you, Ms. Lugwawa. A question for her? I don't know how am I going to do it. We have two questions. Just quickly.
Thank you for the nice presentation. I see on your last graph, you talked about chilling injury.
Yes.
Like you introduced a new variable, if I can say variable, yeah.
Yes.
Of which you never mentioned about it from introduction, background, or anything. You just introduced it on your results. And it's even on the graph, meaning that you measured chilling injury, but you never mentioned anything about it. So I wanted to know, like, did you measure chilling injury or you just assumed or anything of that? Thank you.
Thank you for the question. Well, I did measure the chilling damage. This is because on my methodology, I did mention that the avocados were stored at cold temperatures for 28 days. So this was to simulate the export, simulate the export, it was to simulate export because avocados are transported through ships and it takes longer for them to get to Europe.
All right, thank you. The other question?
Thank you. Out of curiosity, since you are working with avocados, have you ever came across any literature that addresses browning of avocados, and what was the outcome?
Pardon, I didn't get your question. Can you please repeat?
I'm just curious about browning of avocados after you cut them. So I just wanted to check if there's any literature that you came across that addresses that.
Well, no, I didn't, because my study primarily focuses on color change.
All right, thank you. I hope that's answered.
Sorry, I still have a question here. Hi. Yes, I just want to know, of the three, Common Hass is supposedly to be the latest, maturing in December. Maluma is the earliest and Hass is somewhere in the mid. You harvested all of them March, April. Did you do a maturity test on the cultivars before you, because they only mature after picking. They're not mature on the tree yet, so you have to look, did you determine whether they're more or less at the same stage of maturity when they were picked?
Yes, hence, oh, thank you for the question. Well, hence I measured a dry matter content, which measures maturity. And my results showed that there was no significant differences among the 3 cultivars in terms of dry matter. That indicated that they were harvested at a similar period, which in this regard, they were harvested at early season.
All right, I think we can take those conversations to afterwards. Please give Kagisho a big round of applause. Thank you. All right, we have the next speaker, Moripadi Kawa. Would you please join us? They'll be talking about nutrient composition and metabolomics properties of the algae as potential feed for bushveld chickens. Please give her a round of applause. Thank you. Just as a reminder, Moripadi, just when you are looking, just look towards me. I'm going to hold up these for you, okay?
Okay, thank you. Which one? This one? Okay. Good day. Standing before you is Mudipadi Ostasia. I'm a master's student under the supervision of Prof. Manielo, and I'm here today to present under the topic that says nutrient composition and the metabolomics properties of the algae as a potential feed for bushveld chickens. Displayed is the outline of the present study. I'm gonna take you through the background. Indigenous chicken play an important role in a rural community, especially the socio-cultural life and the empowerment of women. Poultry feeds cost around 70% of the output due to the high price of protein supplements such as soybeans, thus having a negative impact on the poultry production around the globe. Algae are high in protein, vitamin, pigment, lipid, essential minerals, carbohydrate, and polyunsaturated fatty acid. They are alternative source of protein that are accessible, affordable, and need to be employed to tackle the issue of high feed cost as commercial and small-scale poultry farming is expanding. According to this author, algae supplementation in the diet improve the intestinal microbial balance, resulting in better digestion and nutrient absorption of broiler chicken. Therefore, the study aimed to investigate the nutrient composition and metabolomic properties of the algae as a potential feed for bushveld chickens. Displayed is the material and method. The study was conducted at KwaZulu-Natal Agricultural and Rural Development, SIDERA, and University of Stellenbosch Central Analytical Facility Laboratory. Algae were harvested from the Oostblue Lepalala Berries Farm, delivered at the University of Limpopo, dried and stored under dry and cool storage for 1 week until they were analyzed. Samples were analyzed based on the dry matter basis, including the ash, the crude protein, moisture, NDF, and ADF carbohydrate, following the AOAC 2012 method. Secondary metabolamide sample were analyzed via the LCMS method using the water synapse G2 QTQF MS connected to a water acuity
UPLC for high resolution UPLC MC analyzers. The data was analyzed using the SAS 2022. Displayed is table 1 showing the nutrient composition of algae and nutrient and the chicken requirement. As we can see displayed, the crude protein, the ADF, the NDF, as well as the ash content, percentage are high compared to what the chicken requires. Displayed is the table 2 showing the mineral composition of algae and chicken mineral requirement. Highlighted in red are the minerals that obtained high content as compared to what the chicken is required. Displayed again is the table 3 showing the phenolic compound identified in algae using the LCM analyzers. As we can see, the code 252-03464 highlighted in red shows the high MSE fragment, followed by the 1,6-digolyl beta-D-glucopyranoid, and then we displayed in blue shows the alpha-dimethylcholic acid with a high retention time. Displayed again is the table showing the quantified individual phenolic compound in plants, whereby the highest in mg per kg is the alpha-dimethylcholic acid, followed by the gallic acid. Displayed is the figure 1 showing the chromatograms of algae with the retention time and the total score. Displayed is figure 2 showing the stacked bar graph of phenolic compound showing the highest in the alpha-dimethylcholic acid with the code of 25203464 in the lowest. Displayed is the hierarchical clustering showing the result whereby still the alpha-dimethylcholic acid is still high. As we conclude with the discussion, the finding in the study indicate that green algae have appreciable amount of minerals due to their high crude protein, ADF, NDF, and ash content, which were consistent with the study conducted by this author in 2011. The present findings are consistent with the study of this author displayed, who reported that the green algae are rich in both macro and micro minerals as required by the chicken. The study conducted by this author in 2018 and this other author displayed in 2019 reported that phenolic compound protect against oxidative stress in chicken, reduce inflammation, immune boosting effect, and improve digestibility health. However, this author reported that the dietary supplementation of alpha-dimethylcholic acid showed the improved growth performance, enhanced jejunal morphology, and promoted intestinal health. The supplementation of phenolic compound improved the chiral microbial composition and increased the mitochondrial activity in broiler. The conclusion of this study, it is concluded that algae have appreciable amount of valuable nutrients and diverse phenolic compound. The phenolic compound identified include the phenolic acid and the flavonoid. This highlights algae potential for enhancing immunity and gut functions in chickens. I would like to acknowledge the University of Limpopo supervisory team, the NRF, and the team that helped me from the Lepalala Oz Blue in harvesting the algae. These are some of the references used in the present study. Thank you.
Thank you, Moripadi. Questions from the floor, comments, suggestions for improvement, anything?
Thank you, Moripadi, for an insightful talk. My question is, but first I'll give you a scenario. You know, algae thrive well in polluted water, and they can have anti-nutritional factors such as heavy metal accumulation, which if you feed them to your chicken, they can negatively impact your chicken. So my question is that, did you account testing for the anti-nutritional factors in your algae? Thank you.
I want to get the last part.
Did you account testing for anti-nutritional factors in your algae? Oh. Yes, thank you.
Yes, that we also tested as we sending them for the analytical procedure. They were also tested, but from where we harvested, we found none. Thank you.
Thank you so much. Give a round of applause. Alrighty, next up we have Cedric Seloane. Cedric, are you around? Yes, okay, cool. So he'll be talking about post-mortem glycolytic metabolites and shelf life properties of chevon from goats slaughtered using indigenous methods in South Africa.
Thank you so much, Chairperson. Good afternoon, everyone. Standing in front of you today is Seloane Cedric, one of the aspiring mid-scientist student who's still studying at the University of Limpopo. Today I'll be taking you through my presentation titled Post-Mortem Glycolytic Metabolites and Shelf Life Properties of Chevon slaughtered from goat slaughtered using indigenous slaughter method in South Africa. This is the outline of the presentation I'll be following through. Indigenous slaughter, as we all know, it's never going to end, and not anytime soon. So this comes to the attention of saying it increases concerns in many developing parts of the country due to slaughter without prior stunning. The implementation of slaughtering animal requires that animal must be properly stunned before the slaughter procedure can commence. However, in resource-limited households, this is never the case because they actually not aware or they don't have any idea about the implications that stress may cause to the meat or the shelf life of the meat when they are slaughtering the animal. So when you slaughter animals, you actually allowing for improved glycogen in the muscles as energy reserves, which can be used as a target that influence the shelf life of the meat. Hence, we are saying now, rendering goats insensible to pain at slaughter could help optimize energy reserves in these muscles, which I just spoke about the glycogen as that energy reserves, which can therefore extend the shelf life of goat meat. So, and yes, we can agree that actually assessing a slaughter method will help scholars, policymakers, as well as these resource-limited farmers to select method that can actually improve the shelf life of goat meat. So the aim of the study was to determine the post-mortem glycolytic metabolites and shelf life properties of goat meat from nondescript goats slaughtered using indigenous slaughter method, which are, yes, done in South Africa. So the objective was to determine the effect of transverse neck insertion, suprasternal notch piercing, and chest floor piercing, which are the three slaughter methods we'll be talking about on these glycolytic metabolites and the shelf life of goat meat. So it was hypothesized that all these three slaughter methods are actually the same or were the same. So the study was conducted at Senwabarwana, rural village of Bochum, and then we used 18 goats, which the class was adult male castrated goats, and then the slaughter methods were the three slaughter methods I just explained, the TNI, SNP, and the CFP. The slaughter procedure was done in the morning around 7 a.m., and then it was done by well-trained and experienced slaughtermen. So how the goats were managed, they were managed on free-ranging communal rangelands. And then before we could actually slaughter the goats, we held them in larages 16 hours with access to water, but we deprived them feed. And then the slaughter practice was done following the study of this author published in 2020 and 2021. So the protocol that was followed was the hazard analytical critical control point. And then through the experimental design, we used a random selection to allocate our goats into three different slaughter methods. For stratification, we used the live weight as the core variate, and then mixed effect model with 2 fixed factors and 1 random factor was used. The Chevron shelf life was the factor under evaluation. So this table basically just explain or summarize what I was just telling you, where we had a factor and a variable which we were determining. And then for measure unit, we were focusing on the longissimus muscle, and then the period of shelf life, where we were analyzing the shelf life, it was day 1, and then again at day 3, 7, 11, and lastly, day 14, post-mortem shelf life analysis. So the next table is summarizing the variables which we determined and the procedure which was used to determine those variables. And then we looked at the glycolytic potentials, where we were focusing on the glycogen and the lactate which is produced. And then so on the right, it explained how this method was done. The other thing we looked at was the pH, as well as the color parameters, which include the redness of the meat, the yellowness, the lightness, the chroma value, and the hue angle. So for data analysis, we use SAS 9.4 version of 2017 software. The GLM, General Linear Model procedure, was used with two-way ANOVA and with interaction and a covariate, as I explained the live weight, initial live weight as the cover rate, to determine significant factors, 2 factors under evaluation, which was the shelf life and the glycolytic metabolites of the goat meat. So the least square estimate, we used mean separation for Fisher's LSD. The significant level alpha was set at 0.05, and then the statistical model displayed below is the one we used, where the YIJ was the response variable, of course, and then the U was the population mean, the SI was the effect of stunning method, either one of the 3 slaughter method, and then the T was the time, and then we had an interaction between time and stunning slaughter method, as well as the live weight, which was the cover rate and the residual error. So, taking you through the result, the first table is for glycolytic metabolites which were analyzed. And then if you can check at pH 24, that's where we had a significant value between these 3 slaughter method. And then for SNP slaughter method, if you can check, the pH was very high as compared to the other 2 slaughter method of TNI and CFP. So again, for glycolytic metabolites, as I mentioned, it was a combination of glycogen and lactate or lactic acid, which is produced in the muscle. And then if you check for the SNP, it had lower glycogen reserves, which is the energy reserves in the muscles. And then as compared to the 2 slaughter method, and then of course, if the glycogen was low, we have to expect the lactate to be high, because lactate is produced at the expense of the glycogen when it's depleted in the muscle. And then also for color parameters, you can see the TNI slaughter method had higher redness values, meaning the meat was more red, or it appeared to be red as compared to the other 2 slaughter method. And then for yellowness, the meat was high for the 2 slaughter method of TNI and CFP, and then chroma, the slaughter method of CFP and TNI also had higher chroma values. So taking you to the next table, where we were actually now looking at the shelf life, how was the meat changing over time for 14 days. If you can check the value of TNI slaughter method, the A, which is highlighted in yellow, I believe you can see it on the table. This value was high as compared to the other 2 slaughter methods. This shows us the meat was a bit redder, or was red in color, brighter red, as compared to SNP and CFP. And we know color plays a very important role when it comes to purchasing, especially to consumers. Before they can actually buy the meat, they buy what they see. Meaning, in other terms, the eyes are what buys before you can actually pop up the money. So for value of yellowness, which is denoted by B, the CFP method had higher values as compared to SNP and TNI. This indicates that the meat appears to be more yellow instead of red. So it was in a way dull, because goat meat it's a red meat. We have known goat meat to be a red meat. So if the meat is not actually red, it gives us an indication that it's not fresh or it's about to be spoiled. And then yes, if you can see the chroma value, it's high compared to the lightness, sorry, it's high compared to TNI and SNP. This again, it can tell us that when we were slaughtering these goats using the CFP, they experienced severe stress, which actually resulted in them trying to cope with the stress. They were, in a way, using their muscles to cope with the pain that was actually inflicted in them. So in a way, we are saying, if the muscles were constrained, these animals the muscles now, they became less compact. And when you display such meat on the surface, the light can pass through. Hence, we are saying now it becomes more lighter instead of being appearing as red or dark red color. So the next diagram is just showing the trends which were happening over the period of 14 days. The first graph is for redness, how the trend were changing between this stress slaughter method. The next one, it shows the yellowness, and then the last table is for lightness. So in conclusion and recommendation, we say goat meat, glycolytic potential and shelf life properties varied with the type of indigenous slaughter. Yes, it's true, we have witnessed that. And then the hypothesis that this stress slaughter method were the same was rejected. So slaughter method indeed do influence the overall meat pH, the bleed out rate, and the ultimate meat shelf life. It affects the ultimate meat shelf life. So goat muscle tissue from TNI slaughter had longer shelf life compared to SNP and CFP. And then we can now say TNI and CFP in a way are actually a better alternative to SNP to improve both glycolytic potential or glycolytic metabolites and the shelf life of the meat. So furthermore, we're saying the use of traditional method for slaughtering goats using TNI on blood metabolites is suggested or is recommended as one of the recommendations for further studies. And then these are some of the references which I used to compile this study. And then I would like to acknowledge the following stakeholders, and this marks the end of the presentation. Thank you.
Thank you so much, Cedric. That was very informative. Any questions, suggestions, comments from the floor? We have one here in the front. We can take two, actually, a little bit ahead of time, because we've got some people who aren't presenting, so.
Thank you, my brother, for the nice presentation. As a meat lover, I just want to appreciate your study, my man, because I've learned something that I never knew, that you need to preserve energy in the muscles of animals for meat to. I only used to know that you put meat in the fridge, you freeze it, and then it can go for 3 months like that. But thank you so much for the knowledge. However, my question is, I wanted to understand if your study is aimed at addressing problems that are being faced at a subsistence level of farming or commercial level of farming.
Okay, thank you so very much for that question. My study actually was targeting the problems that are faced as a subsistence farming, the resource limited farmer. Because we know mostly in rural communities, that's where we find people who, they take their tradition and culture very seriously. So looking at this slaughter method, the reason why they practice them is because they have, firstly, the cultural meaning or the traditional meaning to them. So, however, they still stick to this method because they hold that meaning to their tradition or culture. They practice them, but at the end of the day, they are not aware about the stress that they actually inflict on this animal when they slaughter them. So my study was actually to target those resource-limited farmers.
Okay, thank you. I just want to know if those subsistence goat farmers, are they going to be able to afford the other slaughter methods that you are actually trying to introduce with your study? Thank you.
Okay, thank you again for the follow-up question. Okay, in terms of answering your question, we are actually not saying they shouldn't deviate completely from those slaughter methods to adopt the other ones which they can afford. But then we are actually comparing the three, and we are suggesting to them the best method which they can actually stick to it. If they were to slaughter the animals and they want the meat to last longer. But then if they just want to slaughter and then they don't really want the meat to last longer, they can use the other method. But then the best one which can actually alleviate one of their problems, which is zero hunger, where they will have available food, or to target the food security, they can use the TNI, which was the better alternative as compared to slaughter these goats. Thank you.
All right, thank you. Where's the other question? Do you have the mic with you?
Thank you, Mr. Cedric. I guess I share the same sentiment with my colleagues that this really is an interesting study. Since you said the hypothesis was rejected, that these methods are not the same, so like, can you just briefly explain the differences in these methods and which one can you, like, in your opinion, go with?
Okay, say that again, the last one, which one can?
Which one is the best method, in your opinion, to produce the lovable red color that we want?
All right, thank you so much for that. The reason why we reject the null hypothesis, we're actually saying all the methods are the same, meaning they will yield the same results. There won't be any difference to say actually they are different from each other. But then we observe that they are actually not the same. When you slaughter your animals using TNI, transverse neck incision, that's where the meat tend to last longer as compared to the other two methods. So part of the reason why it's like that, or why that's the case, is because when you slaughter goats using TNI, is the method whereby you just cut the neck completely, right? So when you cut the neck completely, which means you are able to achieve effective bleeding. Hence, somewhere I mentioned that there was effective bleed out rate, right? So if you are able to achieve effective bleeding, meaning you expel more blood out of the carcass of the animal. And then as more blood is expelled, remember blood is another precursor which is responsible for causing microbial, or is favorable for environment of microbes to be present in the animal, and they can spoil the meat. So if you are able to bleed the animal completely, which means there is less blood remaining in the carcass, then that meat, we're saying, okay, it has a potential chance of lasting longer, because as I mentioned, blood is a precursor for microorganisms to survive. Thank you.
Thank you, Cedric. Give him a round of applause, everybody. All right, our third last speaker for the day and for the whole faculty research days is Dimakatso Mayingisane. I'm sorry for the terrible pronunciation. So she'll be talking about optimizing the application timing of biostimulant Calpac using Topsis-Shannon entropy analysis for improving bell pepper sweetness. Thank you, Dimakatso.
Greetings, everyone. I'm Maingisandi Magadzo. I'll be presenting my research titled Optimizing the Application Timing of Biostimulant Calpac Using Topsis-Shannon Entropy Analysis for Improving Bell Pepper Sweetness under the supervision of Prof. Mafio and Dr. Sater. Bell pepper is an important vegetable crop in human diet as it contains various vitamins such as vitamin E and bioactive compounds. Bell pepper is an important vegetable crop in human diet as it contains various vitamins such as vitamin active compounds such as carotenoids and phenolic compounds. Its production is affected by various factors such as pre- and post-harvest
right, I think, Dimakatso, if you want, you can take a moment. We can ask the next person to chat. Otherwise, just take a deep breath. You're fine.
No, I can continue.
You got this. Okay.
Can I start?
Yes, you're good.
Bell pepper is an important vegetable crop in human diet as it contains various vitamins and bioactive compounds such as carotenoids and phenolics. Its production is affected by various factors such as pre- and post-harvest factors such as the environmental conditions and nutrient availability. Seaweed-based biostimulants are natural compounds or substances that when applied in small quantities to plants, they enhance yield, quality, and development of plants. Can I please take a deep breath?
You're welcome. Let's take a deep breath. Can I ask that Puleng, maybe the next person, let's just give Dimakatso a moment. All right, guys, sometimes the nerves get the better of us, hey? Deep breaths. All right, Puleng, are you here? Are you confident? You happy to present? All right, we're going to just give Dimakatso a little time to just take a moment. So Puleng will be presenting about the phenotypic characterization of growth, reproductive, and adaptive traits of stud Brahman under extensive grazing systems.
Greetings, everyone. My name is Puleng Mashamaite, and I'll be presenting a paper on phenotypic characterization of growth, reproductive, and adaptive traits of stud Brahman beef cattle under extensive grazing system. This study was guided by Professor Tada. The study is going to start from the introduction as we go to conclusions and providing recommendations, and lastly, providing references that were cited in the study. Before we can get deep into the problems, the challenges, and how to solve those challenges in the beef industry, I would love to give a brief background in the beef cattle production in South Africa, which mainly operates under extensive grazing system, whereby cattle are raised and normally are maintained on natural pasture as their main feed source. Amongst other beef cattle in South Africa, Brahman breed still remains the anchor to the South African beef industry, whereby environmental challenges and limited resources are common. The breed is known mostly for its adaptability, resilience, its ability to thrive on low quality forages, its resistance to parasites, and also its ability to reproduce in harsh environmental conditions. Extensive grazing system in cattle performance is mostly characterized by the combination of the environment, nutritional factors, as well as management factors. And all of these factors, either combined or sometimes individually, can influence growth, reproductive efficiency, as well as overall adaptability of animal. However, the variation in all of this trait can lead to a significant difference in how well the individual animal, or rather the herd, may meet minimum breeding standards. Other challenges that are faced by mostly smallholder farmers as well as commercial farmers is optimizing the breed performance, especially in Limpopo province, in order to meet the breed standards and ensuring economic profitability. Other challenges normally include climate change, diseases, as well as feed quality and feed unavailability. Despite the breed's adaptability, or rather its ability to adjust to changing environmental conditions, there's a need to optimize the breed productivity, which will ensure sustainability as well as profitability of the beef enterprise. Normally, when we have an increased population as well as increased income level, you'll find people having a high demand for good quality protein as well as the premium meat. Normally some would say the Woolworth premium meat. Another challenges we have climate change, which as a result will influence the landscape, the adaptive traits, which normally contribute to heat tolerance, disease resistance, as well as the efficiency, are now becoming more critical, which as a result, mitigating all of these factors can help the enterprise to meet the breed standard as well as ensuring economic sustainability. The study evaluated trait performance of stud Brahman breed cattle raised under extensive grazing conditions in Limpopo province, whereby the study determined the following objectives: characterizing the growth, the reproductive, as well as the adaptive traits, also comparing these traits from farm level with the national breed standard, and also determining factors affecting all of these traits of the Brahman cattle. As we move on to materials and method of the study, this table here shows the study site, which was an enterprise in Limpopo province, Waterberg district, and our breed, it's Brahman breed, which would normally vary in color, shown here on figure 1, which shows a bit of light gray, and on figure 2, it's reddish, and figure 3, it's black. The sample size was 224 stud Brahman beef cattle. Factors that were evaluated include season, sex, as well as year of birth, and variables that were measured are the traits, growth traits, reproductive traits, and adaptive traits, following a longitudinal observational study. The animals in this study were actually managed conventionally, whereby the husbandry practices that were included were in the standard form, following the conventional method of managing animals in commercial enterprises. The study used a Minitabs 18.7 software of 2017 to analyze the data, whereby procedures of t-test were used to compare the observed value in a farm level as to the national breed standard. And the model that was also followed as there, we also have used the ANOVA, which were used to determine the least square means for fixed factors, and also followed the Turkish HSD test to determine the 5% significance level. On the results, the first table, table 2 and table 3 shows the results of sex on growth performance traits of Brahman beef cattle. For birth weight, weaning weight, yearling weight, there was no significant difference. But however, for average daily gain weight, male performed higher than the females, whereby, however, on average daily gain weight from yearling to 18 months, as well as from 18 months to 24 months, the females outperformed males. Normally, or rather in general, males are known to perform better than females, especially from birth, in order to accommodate their hormonal changes for muscle growth, whereby the females from yearling, that's when they are starting for breeding seasons. So the increase in average daily gain weight basically attributes for physiological changes. We also have the effect of birth season on live weight performance. I have two tables for this. We have the other one which shows non-significant difference. And we also have from 18 months yearling weight during the summer season and spring season, we have experienced higher weight, which is normally attributed by seasonal changes. And then in summer season and spring, normally this seasons experience higher rainfall, whereby there's availability of palatable forages. We have effect of best season for tick infestation, whereby in summer season, there was higher tick infestation and lower tick infestation in winter, which are normally described by tick starts increasing when there is high rainfall as well as favorable weather conditions, which includes high temperature and humid areas. On this graph, we have birth year on different growth traits of Brahman cattle. We have weaning weight that was declining from 2014 to 2016. This decline is normally described by drought season. In 2016, there was a drought that was affecting South Africa, including Limpopo as one of the other provinces. Here we have birth year on reproductive parameters, which is age at first calving as well as calving intervals. For calving intervals, there were non-significant difference, except from first to second parity. There was a significant difference, which is explained normally by maturity of the breeds. Then we also have comparison of average growth parameters with the breed standard, whereby birth weight and weaning weight were higher than those of the breed standards. And on comparison of reproductive parameters of breed standard, we have incidences of dystocia, abortion, as well as weaning rate that have improved and were better compared to those of the national standard, as well as the higher scrotum circumference. However, calving survival rate was lower, which may have been influenced by stillbirth incidences. We also have adaptive parameters. They were also higher compared to national breed standard, showing that the breed was adapting to the environment. In conclusion, the Brahman breed is well suited for the extensive grazing system with good adaptive as well as reproductive traits, which were shown by exceeding the national breed standards. The growth performance, yearling weights were lower, and there were seasonal variation that were attributed by increased tick infestation in summer season. And therefore, I think I would say it is safe to advise farmers to, when it comes to time of controlling tick infestations, they would rather rotate opt for rotational grazing, as well as rotating the vaccines that are used to prevent vaccine resistance of the ticks. And also, they should opt for selective breeding in order to ensure continuous improved reproductive traits, and also opt for specific sex feeding strategies in order to meet the physiological changes of the females and the males' breeds. Thank you.
Thank you so much, Puleng. Any questions, comments, suggestions from the floor? There's another one from our regular customer, it seems, today.
Thank you, Ms. Puleng. That was an insightful presentation. My question is basically around the breed standards. Why was it important for these animals to meet their breeding standard, and how are you sure that they have met these breeding standards? Thank you.
Okay, thank you for your question. Normally, since the Brahman breed as a breeding beef cattle, and also it's used in auctions. So the first reason for that, for it to meet the standards, is to preserve its integrity. And by sticking to these standards, we are also trying to reduce depression, inbreeding depression, and we are also trying to reduce incidences of diseases, and we are also trying to increase the genetic pool. And the last question, I would say by management, proper nutrition management, as well as being able to adjust with seasonal changes and other varying factors.
Thank you, Puleng. I hope that answered it. Alrighty. Thank you so much. Let's give a round of applause. Sorry. Okay, another regular customer on this side for a question.
Okay, so it's not really something that you talked about, but just out of interest, second engagement purposes, I've always wanted to know what that hump at the back of a Brahman, what is it for, what purpose does it serve, and does it play any significant role maybe when breeds are being, the best performing breeds are being chosen, and yeah, something like that?
Yes, the hump does play a very big role because it's an adaptive trait. That hump assists the Brahman beef in adapting to harsh environmental conditions.
Alrighty, let's give a round of applause. Thank you. Our second last person, Tabang Sako. He is presenting on the structural indices of the Bapedi sheep reared under extensive management systems in Limpopo province. Let's give a round of applause, please.
Greetings everyone, I'm Tabang Sako. I'll be presenting about the structural indices of Bapedi sheep that are reared under extensive management system where that happens in the Limpopo province. However, I'm under the supervision of Prof. T.L. Jasi and co-supervision of Prof. J.W. Ngambi. So this is the presentation outline. So the Bapedi Oh, sharp. So yeah, the Bapedi sheep, which is a South African indigenous sheep breed, which is known for its toughness, its disease-resistant characteristic, which is characterized by the Fat tail is a breed that originates in the semi-arid regions of the Limpopo province. However, due to numbers being down, this breed is at the risk of extinction. So morphological characterization is of paramount importance in terms of the provision of insightful knowledge on the breed, so that the recommendations on future breeding plans for improvement and conservation can be made confidently so. So with that being said, the type and the function of a breed are considered better indicators of the usefulness of the animal than the body weight. However, this can be done using the ratios of some of the morphometric trait measurements that give rise to several indices, which are popularly known as what we call the structural indices. On the pictorial presentation, figure 1, we have the Bapedi sheep ewe, and then on figure 2, we have a pictorial presentation of a Bapedi sheep ram. Then phenotypically, Bapedi sheep, they have white bodies and brown heads. However, no specific studies have characterized their phenotypic appearances. So with that being said, usage of the structural indices to evaluate the body conformation, assess the animal's weight and type, is actually superior to the usage of individual measurements in terms of because they provide tested empirical values, which are then limited in the use of the single linear body measurements. Therefore, to minimize the discrepancies when coming to breed characterization, the type and the function of a breed, they need to be assessed. So several authors have already investigated the structural indices in different sheep breeds, and they have concluded that the structural indices are better indicators and also useful in terms of animal characterization than the body weight. So the aim of this study was to assess the structural indices for the evaluation of the performances of the Bapedi sheep breed. So with that being said, the objective, it was to determine the type and the function of the Bapedi sheep breed using the relationships between the morphometric trait measurements. So the hypothesis is in line with the objective. So in terms of the materials and methods, or I can say the methodologies, the study was conducted in the Limpopo province. The animals are populated throughout the province. However, as I already alluded, that this breed type is at the risk of extinction. So with that being said, farmers that are registered with the Bapedi sheep breed, which is the society that is trying to work hard in terms of conservation of this breed, is the one that is actually at the forefront of the conservation. So most of these sheep breed farmers are situated in the Sekhukhune district. So in terms of the animals that were used, we used a total of 127 Bapedi sheep, that is 102 ewes and 25 rams. So just like I said, speaking about the Bapedi sheep society, we purposely selected the sheep and used the census approach because of the numbers. So all these sheep, they were managed extensively, so, and a cross-sectional study design was used. The cross-sectional study design is the one whereby data is collected at a single point in time. So with regards to the collection of the data, the study was authorized by the University of Limpopo Animal Research Ethics Committee with that project number. So in terms of the morphometric trait measurements, they were taken in standing position following the guidelines from the mentioned authors. So the mathematic traits that were measured using the Taylor measuring tape were the body length, the heart width height, sternum height, rump width, rump length, and the canine bone circumference. And then with regards to the body weight, we used the weighing scale. So this is the pictorial presentation of the tools that were used in terms of the data collection. Figure 3 is the weighing scale that was used to collect the body weight. Figure 4, Taylor measuring tape for the linear body measurements. So moving on, so since we spoke, already alluded that structural indices are a combination of different body measurements. So this is a table of the structural indices that were actually calculated. For an example, we can use let's use a height index, for example. The height index is calculated using width height divided by the body length multiplied by 100. So you can just see that it's a combination of one or a combination of two or more linear body measurements. So in terms of the meaning of different structural indices, They are explained on table 2, whereby the first one speaks of the body index. The body index, which speaks of the proportion of the animal, whereby it's speaking to the Baronian system of classification, whereby when an animal is more than 90, it is regarded as longy line. And if it's between 85 and 89, it's regarded as medic line. And then if it's less than 85, it's regarded as a brevic line. And this will tell you the type, whether this animal is for meat, whether this animal is for milk. And then with regards to the statistical analysis, data was analyzed using SES, and for descriptive statistics, the procedure for means was used, and for differences between the different ages, the procedure for analysis of variance was used. And then in terms of the results, with regards to table 1 is the results from the use. As you can see, with regards to the use, I don't know what happened there, but then things happen. So with regards to the differences, you can see that with regards to the height index, the length index, the cephalic index, as well as the compactness of the animal, you can see that there was significant differences. And then in terms of the RAMs, the only differences were seen for the compact indexes only. My sincere apologies, I don't know what happened there. But then moving on, with regards to the discussion, our results, they show that Bapedi sheep is of medic line, given that it is between the ranges of 85 and 88. So this type, it was in close accordance with the study that was conducted in 2017, and also they are lower compared to the study that was conducted in 2021 in the indigenous sheep of Ethiopia. So with that being said, the Bapedi sheep falling between the medic line tells us that the Bapedi sheep are actually the meat-producing animal, because they fall within the ranges that, in terms of proportionality, that is closer to being rectangular in shape. And then with regard to the length index, the results are in close accordance with the study conducted in 2017 and higher than the ones that were in Uda sheep that was conducted in 2018, which tell us the Bapedi sheep have a relatively higher length index. So a relatively higher length index means that the animal is long line. Most of the time, you find that the long line animals also are meat animal. So the results further on tell us the body index ratio are similar to the ones of the study that was conducted in 2021 in indigenous sheep of Ethiopia. However, this body index ratio will tells us that the wither's height is lower compared to the ram height, which basically tells us that the hind region is taller than the fore region. And then in terms of the thoracic development, a good thoracic development is above 1.2. And then the results gave us a thoracic development of ranges between 1.5 and 1.6, which basically means the Bapedi sheep have a well-developed thoracic development, which then enables them to walk for longer distances. And then the results of the study told us that the compact index values were way below 2.60. So therefore, the lower compact index values mean that the animal is a milk type of animal. So this shows us also, as much as Bapedi sheep is a meat type of animal, it can also give us the milk. So the findings of this current study, they reported extremely low ductile thoracic values compared to the findings of the study in 2021. Therefore, this basically means this gives the animal the ability to walk longer distances and also to produce the milk. If you can see the udder of the Bapedi sheep, it's actually seen that as much as it's not that big compared to the other milk-producing animals, it can produce quite fair amounts such that people can consume. So the results of this current study also indicate that similar pelvic index values, same as to the ones that were reported in 2021 in indigenous sheep populations of Ethiopia. So these results reported higher pelvic index values. The higher pelvic index values, they mean the animal will have a wider birth passage. So wider birth passage will literally mean that the animal will not have any problems when coming to dystocia. So Bapedi sheep are actually less prone to dystocia. So in conclusion, This study concludes that with regards to the body conformation of the Bapedi sheep, it can be described as medilinear, which means it's closer to being rectangular, and the better conformation means that the sheep breed is ideal for meat production. Furthermore, these findings, they indicated that the Bapedi sheep is a light animal that can walk long distances. Therefore, it is less compact and also have a well-developed thoracic area. In terms of their alignment, they align towards the front, and they have a wider birth passage, which basically means they are less prone to dystocia. In terms of recommendations, it is recommended that more studies be done in terms of extending the research to molecular level in terms of solidifying the characterization of the Bapedi sheep. So further investigations are needed in other indigenous sheep breeds of South Africa, because in terms of comparison, we use sheep breeds of Nigeria, of Ethiopia, and all those things, while we can actually compare with the ones in our close origins. So in terms of the acknowledgements, I would like to thank the University of Limpopo Department of Agricultural Economics and Animal Production for allowing me the chance to study, the Limpopo Department of Agriculture and Rural Development, as well as the Bapedi Sheep Breeder Society for allowing this study to continue in terms of conservation or characterization of this precious sheep breed referred to as the Bapedi sheep. My sincerest acknowledgements and heartfelt to Prof. T.L. Jassy and Prof. J.W. Ngambi, and last but not least, the Jassy research team. So with that being said, these are some of the references that were used in the study. Thank you.
Thank you so much, Mr. Sako. All right, questions, comments, suggestions?
Sorry, you must just excuse me. I'm not so familiar with the animal research, but when I looked at both this title and the one before, my first incline was to think it was going to be a comparison between external, what do you call it, extensive and intensive grazing, intensive and extensive management, because it's almost like organic growing in plants as well. But now it's more like a characterization of that breed. So it was just that the titles was a little bit confusing for me, because I'm still waiting to see how does this now compare to an intensive management system. I was of the thinking that maybe the extensive management system is changing the way that the animals are developing, because they are managed different and grazing different. So it's just, for somebody that doesn't know your field, it can be a little bit confusing. So are you going to investigate that as well, or is it not an objective?
Okay, I think going forward we can put that into motion, given that now the whole idea is to actually try to record this specific animal. Like, we want to understand what is its average body weight at around about this age. What is it? What can it be bred for? Remember, now the whole idea is to conserve this specific breed of animals. So in order for you to conserve it, you have to know about it. So knowing about it makes it simple for you to be able to compare it to the others, because now we are at a point whereby most of the breeders are using exotic animals, your dorper to try, and your bosfelters to try and make the bigger animals and all those things. So the idea is to document this breed, and after documenting it, then we can see how best can we improve it to try and compete. So now going back to the title. The title, the reason why it was specific in mentioning the extensive environment is was just to put it out there to show the management that was actually used. I understand where you're coming from in saying that why not speak of the intensive or why and all those type of things. So from this, then we can actually try to consider keeping it intensively and actually getting to compare the different environments and see how best it can or how it will perform. Thank you.
All right, let's give him a round of applause. Thank you. Thank you, Tabang.
Okay, thank you.
Alrighty, Dimakatso, have you got the strength for this? This is a learning experience for all of us, so don't be hard on yourself. You've got this. Let's give a big round of applause, guys.
Greetings everyone, I'm Maingisandi Magato. I'll be presenting my research titled Optimizing the Application Timing of Biostimulant Galpac using Topsis-Shannon Entropy Analysis for Improving Bell Pepper Sweetness under the supervision of Dr. Satechi and Prof. Mafio. Bell pepper is an important vegetable crop in human diet as it contains various vitamins and bioactive compounds. Its production is affected by various pre-harvest factors such as climatic conditions and nutrient availability in the soil. Biostimulants are natural substances that, when applied in low concentrations, increases or enhance plant development and produce quality. The decision to purchase fruits is dependent on the internal quality and flavor of fruits, which are related to sweetness of the produce. Therefore, it is important to determine and quantify bell pepper sweetness. Production of bell pepper is heavily dependent on synthetic fertilizers, which pose a significant risk to human health and the environment as a whole. Therefore, farmers are challenged to come up with sustainable technologies such as Galpac. Galpac has been used in various crops to enhance their quality, such as bell peppers, tomatoes, and other agronomic crops such as soybean and maize. However, in those studies, they mainly focus on the concentration, and it's been stated in literature that the efficacy or the effectiveness of these biostimulants are affected by various factors such as application timing based on crop phenology and concentration and crop species. The optimum application timing of Galpac on bell pepper sweetness is unknown. Thus, this study was conducted to optimize the application timing of biostimulant Galpac for improving the sweetness of bell pepper. There is a high demand for produce produced organically because now consumers are more aware of the long-term effects that synthetic fertilizers pose on their health. Such farmers have opted to using seaweed extracts. However, it is important to apply these extracts at the right plant developmental stages to ensure that these plants receive all the necessary nutrients they need for development. As such, we need to come up with analytical tools which will use to optimize application timing, such as Topsis-Shannon entropy. This method evaluates and ranks alternatives in a multi-criteria decision-making problems. It helps you select a best alternative which is closest to the positive ideal solution and furthest from the negative ideal solution. The aim of the study is to optimize the application timing of biostimulant Galpac for improving sweetness of bell pepper. The objective is to determine the optimal application time of biostimulant Galpac using Topsis-Shannon entropy for bell pepper sweetness. The study was conducted at Aquaculture Shade House. The study was laid out in a complete randomized design with 6 replicates, treatments being 5 application times being 1 day before transplanting, 14 days after transplanting. Third, a combination of 1 day before transplanting plus 14 days after transplanting plus fruit set. Fourth, a combination of 1 day before transplanting plus fruit set. Fifth, being a combination of 1 day before transplanting plus 14 days after transplanting, and 2 weeks or 14 days. You are supposed to be 2 weeks before harvest or 14 days before harvest. A refractometer was used to determine total soluble solids, and the titratable acidity was determined using titration method using the titration method. And TSS over TA ratio was also determined by dividing the values of TSS with the values of TA. And also lastly, BREAM was determined using the stated equation. These 4 parameters were chosen because they are the ones which we can use to quantify sweetness or sugars of bell pepper. The data was subjected to gene stat at 5% level of significance. Topsis-Shannon entropy analysis. These are the steps that were followed for the analysis of Topsis. These are the calculations. Results. As indicated in the graph, the third application time in terms of numbers, it recorded the highest TSS ratio, TSS content. However, it was not significantly different from the fourth and fifth application time. Second graph shows the effect of biostimulant application timing on TA percentage. As shown in the graph, the second application time, which is 1 day before transplanting, recorded the highest TA percentage, and it was significantly different from the other application times. The third graph shows the effect of application timing on TSS over TA ratio. As shown in the graph, the third application time recorded the highest TSS over TA ratio, and it was significantly different from the other application times. The last graph shows the effect of application seaweed-based biostimulant GALPEC application timing on bream content. As shown in the graph, the third application time in terms of numbers, it was slightly high. However, it was not significantly different from the other application time, which is the fourth and the fifth application time. So in order to make these results could be attributed to the role of seaweed-based. This result could be attributed to plant growth hormones found in seaweed-based biostimulants. These hormones have shown to improve. These results are in line with. To make sure that we make an informed decision, we then use the Topsis-Shannon entropy as stated in the methodology. The Shannon part of the Topsis-Shannon entropy is used to calculate the weights. Looking at the weights, these are the numbers that we got using the formulas that I showed under methodology. And as shown, the highest weight was recorded in TSS over TA ratio, meaning that TSS over TA ratio is the parameter that has the highest influence in terms of sweetness of bell pepper. After this step, we use the weights that we got to finalize the calculations and get the SI positive solutions and SI negative solutions. SI positive solutions indicate the ideal point, the values for ideal point from positive solution, and these ones, they indicate the negative distance from the negative. These ones, the SI negative indicates the distance from the negative ideal point of position. So after this, we're going to use the SI positive and SI negative to calculate the distance, or CI, which is the distance from the ideal point. Our ideal point in this case is to get high sweetness in bell pepper. As shown, after getting the CI, we then rank the numbers that we get from the lowest to the highest, and in our case it showed that the highest CI, it was recorded in 1 day before transplanting plus 14 days after transplanting plus 2 weeks before harvest, which was 1,0. This indicates that this is the application time that we should recommend to farmers to use in order to increase sweetness of bell pepper. Conclusion and recommendations. GALPEC could be used in organic olericulture to enhance bell pepper sweetness, and also the Topsis-Shannon entropy analysis method was able to optimize application timing of GALPEC. Under the under the materials and method, I forgot to mention that the concentration that we used was 0.4%, which is the recommended one. So, from our results, we therefore recommend that Calpec 0.4% should be applied 1 day before transplanting and repeated at 14 days after transplanting and 14 days before harvest for improved sweetness of bell pepper. This is important for farmers, especially poor results due to the reduced application. This helps in terms of reducing the application times, because in most studies, they use these intervals. You find that in a particular farm, they apply more than 7 times or 6 times. So our study helped in terms of like reducing application times, because there is no use applying a certain treatment 7 times while you can get the same results from applying 3 times at the most crucial life stages of a crop. And also this is important for consumer preference, because in the markets, consumers now prefer goods with good internal quality, and sweetness is part of internal quality. I would like to acknowledge the University of Limpopo for giving me a chance to do my study, and the NRF for funding the study, and my supervisors for the support they've given me throughout the study. These are the references that I've used to compile this presentation. Thank you.
Thank you, Dimakatso, conquering all your fears in this presentation. So, any questions for her while she's here? Yes, Doc.
I really liked your presentation because I'm one of those consumers that likes it sweet. But what I want to know is, when I walk into the shop, I don't know whose produces peppers I'm buying. So I won't know whether they applied Calpec. So that won't be a marketing factor at all for the producer to put it on the market to say, oh, but I've got sweeter peppers because of. I think your selling point will be maybe the saving on fertilizer or the costs to grow it, and making it a more affordable, healthy product. So I think you should include that maybe in your study as a side thing to look at, is what is the economics of this and how can it be used as a marketing strategy so that you can entice the farmers to adopt your very heartfelt research.
Thank you.
That's a very nice suggestion. Is there anyone else who would like to add one more thing before we finish off for the day? In the back there. Okay, sorry, there's another one here.
Thank you, Dimagazo, for your presentation. I just want to make a comment on your research design, whereby you are saying that you used a CRD, completely randomized design. And I believe that completely randomized design is strictly used under conditions that are homogeneous. So, especially laboratory conditions where there is no variation. So, I suggest that maybe you should have used the randomized complete block design, whereby you will be blocking variation from factors such as light and wind or air maybe. So yeah, that's all from me. Thank you.
Can I answer that? But my study was conducted under a shade house, so the environmental conditions were the same.
Did you monitor them?
Monitor them how?
The temperature maybe?
If the temperature is high, all the plants will be subjected to a high temperature. If it's low, all the plants will be subjected to a low temperature.
But then there's a fluctuation of temperature.
Within a shade house?
Variation, yes. I believe so.
Okay, I'll look into that.
Thank you.
All right, we'll take the one last question. I think he's very eager.
Thank you, Dimakatso. I just wanted to understand why you chose 0.4% Calpec and not 1% or 2%.
Oh, thank you for the question. Firstly, biostimulants are known to contain plant growth hormones, and we know that plant growth hormones, they function well at low concentrations. And also, as I've already mentioned, that it's a recommended one from previous studies. you.
Hello once more. We have arrived. We have landed very safe. He's saying finally. You all did well, you presented well, we learned. Thanks to you for being good audience. Thanks to all the chairpersons from Tuesday. You did well, although sometimes you allow people to go on and yeah, but all in all, we all did well. So part of the conference really is for us to interact. I've seen some of you with the person you arrived with, you don't talk to any other person, you don't even say hi to others, you are following each other. Yeah? Have you made friends? That's the whole purpose of us coming here, right? Make new friends and even maybe get people from other departments to assist you, interact. If one professor asks you about something here, it is your responsibility, not today, not tomorrow, some other time when you at your own time, you visit his office or her office. You said, no, you asked me about what this and that. Maybe I need clarity. That's the whole purpose of us coming here. Even supervisors is for us to work with each other. So I guess you have made those kind of contacts. If not, you have your booklet, you know who presented what information you can get from whoever. So that's really part of the whole purpose. So tomorrow is our gala dinner. I guess you are all going to be on Dress to Kill. Look at, now it's for time to relax. We'll be singing to Makazi, Samanyalo, and the like. So stress is now, must be out of you. Let's meet tomorrow. The buses, just to remind you once more, we'll leave campus at what time? We said 4, right? So, and we'll leave here once immediately we finish the formal processes or proceedings. Then we all leave. No one is going to look for any other person. So when the bus leaves, we must all be in the bus. Unless if you have money to pay for, then you are no longer our responsibility for your accommodation here. And we left some few cookies outside for you. Enjoy tea. Then next week Monday, back to Milan. Okay, bye-bye colleagues and ladies and gentlemen. Thanks for your time.